I think we'll make a start then in the interest of time. Like I say, a brief introduction, and then we'll kick proceedings off. So hopefully people will be able to join during that period of time. So good morning, everybody. Good afternoon. Good evening, wherever you're joining from us. It's great that we've had such a good response to this meeting, and we've got people from all over the world. So fantastic to see you all. So my name's Anthony Holmes. I'm Director of Science and Technology at the UK National Centre for the 3Rs. It really is a privilege to be co-chairing with Nicole Kleinsroy from NICETM, the MPS Core Working Group, and Nicole chaired yesterday. And we're delighted to be able to bring you this second day of our spring workshop, along with the other board members from the MPS Core Working Group. Clearly, MPS has played a really significant role in our understanding and treatment of COVID during the pandemic. And as highlighted by Nicole yesterday, we conceived the idea for this workshop before COVID was downgraded and no longer categorized as a public health emergency of international concern, because we really wanted to explore how MPS could be applied in other infectious disease areas relevant now, and also what may be lurking around the corner in the future. Next slide, please. So just as a quick reminder, our motivations for establishing the MPS Core really focused on trying to provide a forum for researchers and other relevant stakeholders from across academia, industry, companies developing MPS, regulators and government agencies to come together and share knowledge and expertise and experiences around microphysiological systems and their application in COVID research to help reduce our reliance on some of the animal models, which may be poorly predictive, but also to help accelerate our understanding and treatment of the disease. You are likely already part of this working group, but if you're not, then we were absolutely delighted to have new members join us. So please, if this is you, you can contact Amber Daniel down at the email address that you can see here, and we'll get you onboarded. Next slide, please. So as mentioned already, this is really a collaborative effort between many individuals and many organizations that you can see listed here. I won't name them all, but I will leave this up there so you can have a look. We're incredibly grateful, Nicole and I, for the support that this group has given us in helping to move forward the MPS Core and really to help provide this workshop and the other activities that we've done for the community. And I really must give a special thank you to the support staff that you can see listed here from Innovative, who have done a fantastic job in making sure that these activities get off the ground and are well supported, and particularly to Amber Daniel, who has really been instrumental in pulling together today's workshop. Next slide, please. So we've got a really great lineup of speakers for you today from both companies and international government agencies, and this will really build on the fantastic discussions that took place yesterday. And similarly, we'll finish with an exciting panel discussion, and this will be led by my MPS Core board colleague, Candice Kerr from NIAID. And it's really important that in order for this to be as successful as it can be, that you're engaged throughout. And so we've built time within the program to enable this. So please contribute to the discussions with your comments and questions for the speakers and the panelists as we go through the meeting. Next slide, please. So just a couple of housekeeping points before we move on. I know we're all very well versed in Zoom meetings by now, but most of the controls are accessible to you via the toolbar at the bottom of your screen. If you can't see this, if you hover the mouse at the bottom of your screen, it should hopefully pop up. I think for today, given some issues we experienced yesterday, all videos for non-speakers and panelists have been turned off, but you still will be able to use the raise hand features and the Q&A features. If you want to, please do rename yourself in the participants window using your first and last names and your affiliations so that we know who's on the call and where the questions are coming from. Next slide, please. We want this to be a really interactive and discussive meeting, so please do engage with the speakers. We'll take any clarifying questions at the end of each talk, so you can either post these in the chat, but remember to indicate which speaker you're asking the question of in case we don't have time to ask them in real time, and then the speakers can come back to them during the course of the meeting. Or you can ask them directly by raising your hand, and if time permits, you'll have that opportunity to ask the question live. If there's any problems at all with the technology, or if you're struggling in any way, shape, or form, please let myself or Amber know using the chat function, and we will do our best to address that. And I think that completes all the housekeeping and all the introductory materials. So With no further ado, it is a great privilege and honour to hand over to Emily Richardson from CM Bio as our first presenter this morning. Emily, over to you.
Lovely. Thank you, Anthony. Can you hear me okay?
Yes, we can hear you.
Brilliant. Lovely. Well, thank you very much for the introduction. And I just want to say a really big thank you to the organisers for organising this workshop. It's been fantastic so far and really looking forward to today's. So next slide, please. So we all know drug discovery is changing, and I think particularly that is obvious within the infection landscape. We have things like mRNA, antibodies, cellular therapies, which are obviously much more humanized. And obviously in the past, we've been very used to sort of small molecules, but even those animal sort of traditional testing with animals and 2D cultures haven't been predictive, which we all know. So really, this is where we see that MPS is really coming to a front. So next slide, please. So this is where we want to see the preclinical toolbox in the future, where MPS systems are being used alongside things like 2D cell culture and in vivo, as well as things like 3D organized and in silico technology. And together, what we're really hoping is that all of this will allow much more predictivity of human responses to infection or drugs. So next slide. And there's been a lot of talk about this over these sessions and hopefully more later on today as well. But these are the sorts of things that sort of I picked up in terms of MPS model requirements for infectious diseases, things like ease of use and uptake, use in BSL-3+ labs, the device material, the complexity of the model versus the research question. Can you increase and decrease the complexity? And particularly important for infection models, thinking about that immune cell inclusion, which we'll talk about a bit later. And then obviously we all want high translatability to the human and obviously highly reproducible and robust models. Next slide, please. So the physiomimic system from CM Bio was briefly introduced yesterday by Tyler, but just to show you, we have a single and multi-organ systems. These controllers you can see on the left sit outside of the incubator. The docking station at the bottom sits inside the incubator with drivers where the plates fit into. These slide in and out of the docking station, so they can be removed from the incubator into the MBSE very easily. The plates themselves are constructed with COC, so very low drug binding, and the plates are all open well. So that means it's very easy to access them, to add challenges, take media, take tissue samples, etc. And this means that they're very flexible as well. Next slide. So the models that we have internally, you can see here. So these are the current plates we have at CM Bio and offer to customers. So we have our little liver model on the far left, and the perfusion in this plate goes up and through the tissue. And this forms these really lovely liver microtissues you can see there with the lumen, which really recapitulates nicely the in vivo liver tissue infrastructure. We have our barrier model plate, where basically you have an almost like a 12-well plate format. Inserts fit into these plates, and the perfusion goes underneath the insert. And then finally, we have the dual organ model, which you can see on the far right, which combines the two capabilities of the liver and the barrier model in a 6-well plate format. And this is where you can basically combine your barrier tissue and your liver tissue with an interconnecting pump, which allows media flow between the two chambers. And very importantly, these pumps can all be tuned separately and turned on and off separately. And I'll explain the importance of that in some later slides. So next slide, please. So first of all, I'm just going to talk through our lung models. So we have two lung models within CM Bio, a bronchial model and an alveolar model. And you can see nicely from this slide that the lung MPS tissue forms this really nice tissue over 14 days at ALI, which nicely recapitulates the human architecture and biology compared to that of the static 2D over the same period, which forms these sort of more like monolayers to an extent. So next slide. And we've nicely characterized these models. So here you can see the bronchial model. It forms this really nice pseudostratified epithelium with production of ciliated cells and goblet cells. You can see mucus production over time, and we can quantify that. And also it forms a very good barrier that is very consistent over time. And within these models, obviously, because they're insert-based, we can build up different layers. So here you can see we've done epithelium and endothelium, but you can see it's also relatively easy to then add in additional layers such as smooth muscle or fibroblasts in the future. Next slide, please. And then we have our alveolar MPS. So for this model, we really wanted to concentrate on the differentiation of the cells into type 1 and type 2 cells. And you can see here after 14 days of culture that we have a nice population of 81 and 82. Really important here to focus on 82 cells in particular, as these are very important within infection. And quite often in a lot of models, these cells become depleted as they differentiate into type 1 cells. So it's really important for us to be able to keep that population there. And on the right, you can see that the tissue forms this really interesting and quite beautiful alveolar sac-like structures, which you can see. And if you look to the bottom of that image where you've got some staining, we can see in green surfactant protein, and you might just be able to see on the bottom the endothelial cells stretching along the basolateral side of the insert. So next slide. So because we have circulating flow, we can also think about adding circulating immune cells. So for this reason, we added monocytes in this case because of their importance within lung biology and response to infection. So here we added circulating monocytes in the basolateral side, and then also monocytes into the apical side, where they formed these alveolar macrophage-like cells, which you can see on the right-hand side in magenta. And these are patrolling around the apical side of the epithelium. Next slide. So what we wanted to do was to challenge these models and see whether we could see the differences between inflammatory response between the alveolar and the bronchial models, and also how the models responded differently in the presence of monocytes. And what you can see, I just want to point your attention to the bronchial model, where we see basically no response when we add LPS as a challenge. And this was kind of interesting at the time. And then we looked at the in vivo data, whereas the bronchial epithelia does not express a TLL4 receptor, which LPS is the agonist of. So then when we add in our monocytes, we then get our inflammatory response, where the monocytes are reacting to that LPS and inducing the immune response within the tissue. Next slide, please. So obviously we wanted to use these MPS for COVID-19 research, and we were funded by a grant from Innovate UK to do this. So next slide. So we obtained pseudotyped lentivirus expressing the SARS-CoV-2 spike protein from BEI. As Mark pointed out yesterday, BEI has been a really fantastic resource for us. So thanks to them. So to start off, we looked at the entry receptors within our tissue, and we found and focusing on ACE2 and TMPRSS2. And you can see here that the MPS tissue really nicely recapitulates the sort of expression profiles we see in the different areas of the lung within a human, which you can see on the bottom panel. So the bronchial tissue expressed a higher amount of ACE2 compared to the alveoli, which we see in human. And then TMPRSS2 was expressed nicely across both tissues in the same manner. Next slide. And then we wanted to look at whether this pseudotype lentivirus would actually produce an inflammatory response. And indeed, we did see an inflammatory response to the pseudotype lentivirus later through literature, finding out that this is mainly probably due to the presence of the spike protein. And what we can nicely see that when we add in our immune cells in our alveolar model on the apical side, you can really see that the monocytes or the macrophages are now reacting to the infection of the tissue, and they're clustering around those sites of infection. Next slide, please. So once we'd understood the sort of local infection of the lung, we then wanted to understand a bit more of the systemic inflammatory response. And we know that in really severe cases of infection, This induces sepsis quite often, which is obviously a very big killer in the infectious disease space. And the liver is a really key part of this because the liver mounts the acute phase response. And this is what induces that sort of systemic inflammation. And what you also quite often see is that the liver is one of the first organs to be damaged, and ultimately leading to death in severe infection patients. So if you go to the next slide. So we have a very well characterized liver MPS. So this is all data from a paper that we did in collaboration with the FDA CEDAR group back in 2020, where they basically showed that our liver MPS was able to retain metabolic activity and viability over long periods of time, compared to traditional methods such as spheroids and sandwich cultures. And this data has been shown time and time again. What I particularly want to highlight is that this model has been used in hepatitis B research, particularly there was a paper by a group in Imperial who did basically a similar study where they compared our model to these sort of traditional liver cultures. And what they found was sort of similar in that you could culture these for a long time, which was very useful for the hepatitis B infections because they have this longer life cycle. But also they saw things that they weren't able to recapitulate in other in vitro models, such as detection of cccDNA. So these are really, really useful models in this space. Next slide, please. So in terms of the setup, we have a liver co-culture of hepatocytes and Kupffer cells, which are the macrophage of the liver. We then have our lung epithelial and endothelial cultures, and you can see those in microscopy images on the right-hand side. Next slide, please. So this is where I want to highlight, I said at the beginning, the importance of this dual organ plate is the interconnecting pump. And in these experiments, we really utilized this interconnecting pump. So what we did in these experiments was that in the sort of tissue maturation stage, these cultures would co-culture together. So the interconnecting pump was open. Then when we wanted to infect our lung and look at the local infection, we closed our interconnecting pump and allowed the infection to occur over 24 hours. During this time, inflammation was occurring, cytokines and chemokines were being produced. We then opened the interconnect after 24 hours and allowed the two chambers to mix. And we then looked at the inflammatory response, as well as the tissue health and function over time. So if you go to the next slide. And this is what we saw over the 72-hour infection period. And this is really interesting. So what you can see within the first 24 hours is a peak of inflammation, which is resulting from the lung. We then open the interconnect. We see the mixing of the two compartments, and then we get the secondary peak from the liver, where the inflammation from the lung has reached the liver is then responding to that. You get that second peak, and then you get this exponential increase in inflammation, which seems to be recapitulating that exacerbated inflammation that we see in really severe infection cases. Next slide, please. So what we then wanted to do was understand, okay, if we then add in an immune component, what then happens to that inflammation? Next slide. Okay, and then, so sort of unsurprisingly, when we added in our monocytes, we got an enhanced inflammatory response, which you can see on the left. Apologies about the numbers there, they've gone funny. And then but what was really interesting was when we had our monocytes in there, what we also started to see was results of the enhanced inflammation was tissue damage. And you can see that visually here in the liver, where the scaffold density was decreased, and we're able to quantify that on the right. Next slide. And then we looked at both of the tissues and looked at their function and integrity. And what you can see on the left is the barrier integrity of the lung, where we get a reduction when we have no monocytes, but then we get further reduction when we have those circulating monocytes in there. And on the right, the same thing, there's a non-significant decrease when we have no monocytes in the metabolism of the liver, but then when we add in those circulating monocytes, that then decreases to a significant effect. So then we wanted to know, okay, this is affecting the tissue. Are we actually seeing what we would see in a human? So next slide. So what we wanted to look at was, is this infection and inflammation inducing the acute phase response? So to do that, we looked at CRP, which is a marker of the acute phase response. And what you can see at just past 48 hours is the sort of gradual increase and then the spike of CRP production, which corroborates with the albumin production in the liver, showing that that the liver is reacting in this inflammatory way, inducing the acute phase response, and is also being damaged itself, as we would see in those really severe infection and systemic inflammatory responses. Next slide, please. So I'm going to take you back to one of my first slides and the sort of model requirements we saw for the infectious diseases. And hopefully I've been able to show you that the physiomimic system is really useful in that case. So it's very easy to use, and we can use it in a range of different laboratories. As I said, it's currently being used in a range of BSL-3 labs. We have very good relationships with cell suppliers, and we qualify cells in-house that we can then give to customers and collaborators. So we have a very good relationship with them, which is very important for MPS devices. Our plates are all made of COC, so we know that there is less of a drug interaction risk there. Our plates are all open well, which means it's very easy and flexible to add challenge and then also monitor the system over time. As I showed you with that inflammatory response graph, we can really measure it over time, which is very, very useful in thinking about infection research. And then obviously we can change the complexity of the model based on our research question. And hopefully I've been able to show you that there is high translatability to the human. And finally, I haven't put a tick against the highly reproducible. Now, that doesn't mean that I don't think our model is not highly reproducible, far from it. However, I think it's very important that it's not the MPS providers that are telling you that this is highly reproducible, but it's actually other users of the system and sort of that lab-to-lab variability and user-to-user variability. So next slide. So we have a number of different customers and collaborators currently using our models across the world, but I want to highlight these two in particular with regards to lung and the multi-organ. So the Liverpool School of Tropical Medicine, ICON Group are currently assessing our lung and lung-liver MPS for COVID-19 research, but in the future also other tropical diseases. And the FTA-seeded DARS group, who we have a long-standing collaboration with, are currently evaluating the use of our lung MPS for predicting ADME properties of inhaled medications. So we really look forward to seeing those results in the near future and showing that robustness and reliability of our models. So next slide. So finally, I'd just like to thank the whole CMBio team, particularly Hayley, who did a lot of this work, who's in the center of that picture there. Many thanks to Innovate UK for the funding, and thank you all for watching, and I will look forward to the questions. Thank you.
Wonderful. Thank you, Emily. Really lovely presentation to kick things off today. And we certainly do have some questions for you, and fantastic timing. So hopefully we've got time to get through all of these. But if not, if you could just go through them and type a response, that'd be fantastic. So we'll start at the top. So firstly from Monique Van Hoek. Apologies for my pronunciations if that's not quite right. How can you add infectious agents to the system such that you can get them into the alveolar spaces?
That's a really interesting question, actually. So the way we've been doing it so far is just adding a very small volume to the apical side of the transwell. And that seems, we've also done a lot of studies with drugs and looking at sort of drug absorption and permeability through the system. And we do see them moving through the alveolar spaces. But I think that's a really interesting question. I think something we'd love to do in the future is do more aerosolization applications of infectious agents or drugs in the future to really understand that a bit more, because I think that'll be important.
Excellent, thank you. And then next question from Jiru Wang. Did you add media to the apical chamber? If yes, how much? Are the media the same in the apical and the basal chambers for the lung immune model?
Yep. So all the lung cultures are at air liquid interface. So on the whole, no, usually there is no media in there. Once we induce ALI, we keep it at air liquid interface. When we're adding challenge, as I just said, we're adding that in a very small volume at about 50 microliters, just to allow it to go across the whole of the insert. So the media volumes are not the same in the apical and the basal chambers. Was it in the lung immune model?
The question, yes. Yeah.
Yeah, so the media volumes aren't the same. I hope that answers your question. If not, please do message and I'll try and qualify that a bit more.
Thanks, and I think we've got time just for one more. So from Peter Gaskin, did you try different times to open the interconnect pump?
Yeah, that's a really interesting question. We didn't. I would have loved to have done that. Unfortunately, as I said, this was all funded by a grant, so we were very compacted in what we could do. But that's absolutely something we'd love to do more of in the future. And as I said, that's the joy of the system, is that you can open and close it independently. So being able to really understand that time frame better would be really, really interesting. So yeah, hopefully something we can do more of in the future.
Fantastic, thanks, Emily. I think we'll move on to the next speaker there, but there is a question from Mark Williams focused on monocytes. If you could answer that, that'd be fantastic using the chat. Thanks very much, Emily, for kicking us off.
Thank you.
So next up, we have Dr. Gabriella Welra from the NIAID Integrated Research Facility. Are you there, Gabby?
Hi, yes, I'm here. Can you hear me?
Fantastic. Yes, we can hear you and we can see you. Thanks very much for joining us today, and I shall hand over to you.
Thank you very much, Anthony, and good morning, everyone. My name is Gabriella Welra. I am a veterinarian and a classical virologist here at the Integrated Research Facility. I lead the molecular virology and the microphysiological systems team, as well as the immunology team. And so my background is mostly on animal models and using and developing animal models for the purpose of studying risk group 3 and risk group 4 viruses in high containment. Next slide, please. All right, so I want to get started with the acknowledgements. So none of this would have been possible without my team, which you can see here in the photo. So all the hands-on work that went into generating the data that I'll be presenting today was done by them. They're really amazing. I also would like to thank Jens Kuhn and Nicole Kleinstreuer, because they really set the foundation for us to get this effort off the ground. And Tyler Goralski and Kyle Glover for their input and guidance on protocol optimization, and really everyone else that was involved in this. Next slide, please. All right, so we're the Integrated Research Facility. We're part actually of NIAID at the NIH. We're located within the Division of Clinical Research. The director is Dr. Connie Schmaljan, and we are a national BSL-4 resource for collaborators. So we are not a CRO, but we're open to collaborating with academia, industry, or really anyone who fits or has a project that fits into our mission. And so we're here to facilitate multidisciplinary research to understand, treat, prevent, and eradicate diseases caused by novel, emerging, and highly virulent viruses. So in terms of our space, we have Biosafety Laboratory 2 space, which really is there to support our high containment. And in terms of the biosafety level 4 maximum containment space, we have animal as well as laboratory space. That's quite extensive. We do have a smaller BSL-3 suite, but that is just kind of a limited or temporary use of a BSL-4 base. We have some quite unique capabilities, and that first and foremost includes our medical imaging. So we have PET, CT, and MR. We can image non-human primates or smaller animals such as ferrets, guinea pigs, hamsters, mice. We have aerobiology capability, electron microscopy, animal models, and molecular virology. The viruses that we work with the most is Ebola virus, Lassa virus, Nipah virus, obviously most recently SARS-CoV-2. So essentially any risk group 4 virus technically, and then some select risk group 3 viruses on occasion. Next slide, please. So I wanted to sort of tell you about our MPS capabilities at the IRF Frederick. So we started this effort about 3 years ago. And currently we're using organ chips, and the platform that we're using is from Emulate. In terms of our capacity, we have a system in the BSL-2 and then another system in the BSL-4 with 4 Zoes each. So that gives us the capability of having 48 chips on either side at any one time. The species that we currently use is human. However, we're interested to expand that to use non-human primate and mouse chips. The organs that we've developed so far or implemented is the alveolus chip. That's what we started with, and that's what you can see on the right-hand side depicted. And then we switched to a bronchial or small airway chip. And then in development or in the near future, we want to develop brain chips and liver chips. The viruses that we currently work with are SARS-CoV-2 and Nipah virus most recently, and then hope to transition to more Ebola virus and alpha virus centered collaboration. Next slide, please. All right, so I wanted to include a couple pictures so you can see how our setup looks like. So this is on the BSL-2 side, and I'm sure this doesn't look any different compared to your labs. But yeah, this is my team setting up the chips. So for the bronchial or small airway chip, we typically seed the cells out in BSL-2. We let them differentiate and we subject them to the air-liquid interface. And then once they're ready for exposure or inoculation, we transfer them into the BSL-4 base. Next slide, please. So here you can see how our work looks like at maximum containment. I would say the biggest difference is the personal protective equipment or the PPE. You can see this is a positive pressure suit or the space suit that we wear in a very negative pressure environment. And yeah, but in terms of the chips, everything remains the same essentially. Next slide, please. All right, so for the rest of my talk, I'll be focusing a lot on Nipah virus. So Nipah virus is a paramyxovirus. It is classified as a risk group 4 pathogen. So handling of infectious virus has to occur at BSL-4. The WHO recently classified Nipah virus as a priority pathogen for public health emergency of international concern. I wanted to go over a little bit over the transmission dynamics and the transmission cycle of Nipah virus, which you can see on the top. So the host are Pteropus bats. So these are flying foxes, and spillover of infectious virus can occur really to any domestic animal species. However, pigs appear to be especially susceptible to Nipah virus infection. And from there, spillover can then occur to humans or vice versa. So epidemiologically, outbreaks have in the past occurred in Malaysia, Bangladesh, and parts of India. As I mentioned, it is a zoonotic illness, so there is a chance to apply a One Health approach in solving this overall issue. What I also wanted to point out is that whenever animals are infected or a pig farm is involved, the only solution in those cases is to cull the entire herd. So therefore, these outbreaks cause, aside from the human infections and the public health risk, they also cause enormous agricultural and economic loss and with implications on the trade of that country. So that's important to keep in mind. Human-to-human transmission is possible. That's very important. And there are currently no approved vaccines or therapeutics available to treat Nipah virus infection. And on the left, you can see an EM image of Nipah virus particles that were taken here at the facility. Next slide, please. All right, so in terms of the disease, so Nipah virus has a tropism for the central nervous system as well as the respiratory tract. The disease in humans can be asymptomatic or have a spectrum from mild to fatal. The case fatality rate is about 40 to 70%. It can start off with non-specific symptoms ranging from fever, headache, muscle pain, and then it can progress to acute respiratory infection symptoms that are caused by pneumonia. So this can include cough or dyspnea. There can be neurological manifestations due to encephalitis, such as dizziness, drowsiness, altered consciousness, seizures, and even coma in severe cases. In surviving patients, sometimes there are neurological sequelae that have been described in the past. Now, in pigs, there's also a spectrum of clinical disease. It seems that one particular age group is more susceptible, which are piglets, in which the infection can be lethal. They also exhibit non-specific signs, which can include fever. And what's important is that it may resemble other infectious diseases of swine, which means that Nipah virus infection would not be necessarily on the top of the differential diagnosis list. But it can also lead to labored breathing, barking cough, all signs of respiratory infection, and also include neurological signs such as trembling, twitching, muscle spasms. There's nothing known about long-term sequelae in animals or pigs. Again, that's because, as I mentioned before, pigs get essentially terminated when an outbreak occurs. So we wanted to really see whether we could model Nipah virus infection in a human small airway chip. Next slide, please. So the first effort that we did actually out in BSL-2 is to confirm, really to optimize our protocol and to confirm that we have the right phenotype of the cells that we're using. And so here you can see the small airway epithelial cells and the staining for the cell surface marker expression. So we confirmed that we have the zona occludens, tight junction, E-cadherin. We stained for tubulin and also for proliferation. And on the right-hand side, you can see that the merged images, pre-differentiation and then also post-differentiation. Next slide, please. We did the same thing for the microvascular endothelial cells with a specific staining for V-cadherin, CD31, and you can again see the merged images, pre-differentiation as well as post-differentiation. And what I also should mention is that all of the data that I'll be showing you today, they're new, so they're unpublished. We're currently in the process of wrapping the experiments up and put them in a publication. So I hope you find them interesting. Next slide, please. All right, so we then wanted to, after really confirming, yes, we can, we have the right type of chip, we have the cells, we have the protocol optimized. We then proceeded to see whether Nipah or the chip was permissive for Nipah virus infection. And so what we did is we used Nipah virus Bangladesh. And on day 0 or hour 0, we exposed the top channel that contains the epithelial cells to a multiplicity of infection of 1 of Nipah virus. And then we collected both effluent from the apical and the basal chamber at 24 and 48 hours post-exposure. And we also collected cell lysates and tested those for the presence of Nipah virus. So on the top, you can see on the left-hand side, the Nipah virus genome equivalents per ml, which were analyzed by RT-qPCR. And on the top right, the plaque-forming units, PFU per ml, which indicate presence of infectious virus. And as you can see, there is an increase of the presence of Nipah virus at 48 hours post-infection. And what I actually would like to point out here is when you look at the infectious or PFU data, you don't see any presence of infectious virus in the lower channel or in the endothelial cells, but you do see them at 48 hours post-infection. And so that means that the virus is actually crossing over through the barrier down and infects the endothelial cells as well. We also tested for barrier function, and as you can see on the bottom, and we confirmed that Nipah virus infection actually disrupts the barrier function. Next slide, please. So what we observed, essentially morphologically, that we found interesting is the formation of syncytia among the epithelial cells, and you can see that with the yellow circle, and that's at 48 hours post exposure to Nipah virus. So this is something that is seen otherwise as well. We confirmed that in our chips. And on the right-hand side, this is just to show you that we confirmed the presence of the Nipah virus receptor, which is Ephrin B2 by IFA, pre and post ALI, both in epithelial and endothelial cells. And so this essentially confirms why we see infection in the basal chamber as well, because the receptor is present in both cell types. Next slide, please. All right, so we optimized our protocol. We confirmed that the chips or the cells within the chips were permissive to Nipah virus infection. And so next we wanted to really test whether our system would be responsive to any changes. And we did that by evaluating 2 small molecule antivirals against Nipah virus infection. And so one of them is remdesivir. All of you might be very well familiar with that. So we use remdesivir as a control. It is a nucleotide prodrug of an adenosine analog. So it essentially binds to the viral RNA-directed RNA polymerase and essentially inhibits the viral replication. And remdesivir has previously been shown to be effective against Nipah virus infection in brivets. And then we selected sotaterfen sort of as our test article. So sotaterfen is a selective inhibitor of the eukaryotic initiation factor 4A mediated translation. So it leads to an inhibition of the protein expression, essentially. So it has a different mechanism of action compared to remdesivir. And there's so far no data out that has evaluated sotaterfen in vivo. Next slide, please. All right, so here you can see the plaque assay results of when we introduced Nipah virus to the upper channel only, as compared to introducing Nipah virus together with either sotaterfen or remdesivir on day 0 at the infection time point. So if you're not familiar with plaque assays, it should look like the negative control. There are no plaque forming units. And you can appreciate that at 48 hours, when we only use Nipah virus, there are a lot of plaques. However, the number of plaques is significantly reduced at 48 hours in the sotaterfen and remdesivir treated chip. Next slide, please. So this graph essentially shows, or this slide shows the data that we analyzed, or the effluent that we analyzed from the top and bottom channels that were analyzed by RT-qPCR. So these are the genome equivalents per ml for 24 and 48 hours post-infection. And as you can see, Nipah virus is in orange, and the sotaterfen and remdesivir treated chips are in blue and green. And you can see, especially at 48 hours, you can appreciate a reduction of the genome equivalent or viral copies detected in the effluent. And on the right-hand side, you can see the barrier function, which is disrupted when we have Nipah virus only, but it is significantly improved or intact when we have the presence of sotaterfen or remdesivir. Next slide, please. All right, so the take-home messages here are that we successfully implemented organ chip technology in BSL-4. We confirmed that Nipah virus infects human epithelial and endothelial cells of the bronchial airway chip, and it disrupts barrier function. And we also evaluated 2 small molecule antivirals with 2 different mechanisms of action and showed efficacy against Nipah virus in the small airway on a chip. Next slide. So because this is more focused today on logistical aspects of using MPS, I wanted to include some of the challenges that we encountered using the Emulate platform in our high containment. And there are actually not that many, but I would say one of the biggest challenges is to remove inactivated chips from BSL-4 containment. So currently we can remove cell lysates, we can remove the effluent, but we're currently in the process of removing formalin-fixed chips out of containment so we can actually image them out in BSL-2 because the imaging of those chips in BSL-4 takes a long time. Another challenge I would say is the equipment repair and maintenance, but I have to give a big shout out to Emulate, who has been very receptive and very collaborative and very helpful in making sure that we're able to PM the equipment either inside the 4 or out. Some protocols that might be lengthy or complex may limit the size of experiments because our staff is only able to stay in the containment for so long. So we can either have smaller experiments or it would require more staff. We also require highly skilled staff who have very good tactile skills. You can see in the pictures, we have actually 3 pairs of gloves that we wear. So for small and intricate things that need to be done, there's just a lot of tactile ability that's required. And also just overall the ability to troubleshoot independently while in the 4 is also required. Next slide. Just a few words, so I don't take too much time, on the future applications of MPS that we have sort of in the pipeline. So here you can see the viruses, animals, and organs and MPS styles or systems that we plan on using. They're in no particular order, but we do plan on hopefully having a collaboration that would involve retinoid or retinal organoids as well as brain organoids for the evaluation of Ebola virus, mostly persistent infection, and that's really exciting. We also want to stand up the liver chip, again, mostly to test acute Ebola virus infection. We have collaborators who are interested in working with us on brain chips from non-human primates and mice, mostly for the purpose of alpha viruses and therapeutic testing. And then we also plan to adapt the small airway chip into a porcine small airway chip. Okay, next slide. So I just wanted to mention that we will be participating at the MPS World Summit in Berlin in about 3 weeks. Please come and visit us. A lot of this work has been done and led by Dr. Sushma Bosley, who is here in the picture. I only presented one subset of data. We also used neutrophils isolated from humans by cell sorting, and we introduced them into the channel to then essentially assess the immune response. And so these are all data sets that you'll be presenting on Tuesday, June 27th, again in Berlin, Germany. So please come and say hi. Okay, next slide. And so, yeah, this is really my last slide. I know I'm already at time, but all I wanted to say is if you're interested in collaborating with us, please feel free to reach out either to Dr. Jens Kuhn or myself, and thank you so much for your attention.
Thank you, Gabby. That was a really wonderful talk. Really appreciate it. Just having a look. So there's a question that's just come in from Mark Ferrer. I think we have time to address this certainly. And his question is, did you need stretching for your lung chip work?
So we did stretching. We essentially, when we were optimizing the protocols, we used stretching, but not for our infection work later on downstream.
Okay, thank you. And then just one very quick one from me in terms of a rather silly practical question. In terms of that, having that excellent tactile ability to be able to work in that environment, I mean, is that something that can be learned, or do you just find staff sort of naturally have that ability? How does that work? So I know certainly as soon as I put on anything more than one pair of lab gloves, I lose the ability to use my hands personally. So
Right. Yeah, we have two regular gloves underneath and then a thick glove on top that you could see in a picture. Well, I think whenever we have interviews or we're hiring staff, we already are looking for individuals who have very good skills in BSL-2, and that is typically a promising characteristic that these individuals will perform well at BSL-4. And we have a very extensive training that takes multiple months until these individuals are signed off to enter the BSL-4 independently, and then multiple weeks after that until they can actually do more complex things like manipulating and handling the organ chips in BSL-4.
Wonderful. Thank you. Thank you very much, Gabby. So I think we'll move on. If you have any more questions for Gabby, please do put them in the chat for her, and I'm sure she'll address them over the course of the meeting. So our next speaker this morning is Dwight Yin from NIAID. Dwight, are you there?
Yes, can you hear me?
Yes, we can hear you and we can see you. So I'll hand over to you now. Thank you.
Great, thank you. Thank you for welcoming me. I'm Dwight Yin. I'm from the Maternal Adolescent Pediatric Research Branch in the Division of AIDS at the National Institute of Allergy and Infectious Diseases. NIAID has an interesting setup where we in the Division of AIDS focus on HIV and others such as DMID. Thank you to those colleagues, cover everything else from a infectious diseases standpoint. So we'll be focusing on HIV, and I'll also include some pediatric and maternal aspects given our focus. Okay, next slide, please. So the views expressed in this talk are my own and do not necessarily represent the opinions of NIH, Department of Health and Human Services, or the US government. Okay, next slide, please. So HIV is still a large problem around the world. An estimated 38.4 million people are living with HIV around the world, most of whom are in sub-Saharan Africa, about 20.6 million there. About 1.5 million new infections happen per year, and we have 650,000 AIDS-related deaths per year around the world. Next slide, please. A lot of what we understand about the natural history of HIV originally came from adults. After infection, then you'd have a rise in your plasma HIV viral load over the first 1 to 12 weeks in an acute infection phase, followed by some a stable time for about 6 to 10 years. And then finally, in the last 1 to 2 years, people would progress to AIDS. The CD4 cell count has been the main immune biomarker that has been helping us understand the immune status, which slowly decreases over time until we reach AIDS, which was initially defined as a CD4 of less than 200 cells per less than 200, or having an AIDS-defining illness, such as opportunistic infections, such as pneumocystis pneumonia, or HIV-related encephalopathy, or AIDS-related malignancies. However, in 2006, we understood that there was more than just opportunistic illnesses that we had to worry about as people compared continuous versus interrupted therapy to HIV based on the CD4 cell count, and noticed that people who had interrupted therapy would still develop major cardiovascular, renal, and hepatic diseases, sometimes leading to mortality. So inflammatory diseases have also been very important, and organ diseases have been very important over time. Next slide, please. In children, the story is even a more rapid clinical progression. About 90% of pediatric infections occur via vertical transmission in the perinatal period. Therefore, children are experiencing this very early with an immature immune system. And in this setting, children progress much more rapidly than adults. In fact, the highest incidence of AIDS is in the first year of life. And untreated, about half of children with HIV will die by 18 months of age. And therefore, people were trying to predict which children with HIV needed early therapy versus deferred therapy, and ultimately found that early initiation reduced the hazard of mortality by 76%. So the current focus of the field is trying to find and get as many children on therapy as possible, although only the minority of treatment eligible children receive therapy. Next slide, please. To understand how we can better optimize study of HIV using MPS, it's probably good to understand a little bit more about the virus. Now, HIV is a lentivirus or slow virus, given that it's progressed so slowly as seen earlier in the natural history. But it's a retrovirus. So it starts off as an RNA virus, and then is reverse transcribed to DNA, which can then integrate into the host genome. It can be integrated in the genome in a resting CD4 positive T cell, for example, for long periods of time, but then only when activated do we then get translation and assembly of proteins and budding and maturation. Now, antiretrovirals cannot eradicate integrated DNA, and the host immune response has difficulty sensing the integrated DNA. So what happened was with early sentinel studies back in, say, 1997, then we understood the presence of this HIV latent reservoir, which was, if you can look, even despite antiretroviral therapy, you can still find persistent HIV in the cell. Now, most of these are defective. So you might have some HIV-1 DNA in the cell, and a smaller proportion are integrated DNA, and also a small proportion are replication competent. Many are defective. In fact, most are defective. And therefore, and then an even smaller proportion are persistent replication competent virions. And they may be present in the peripheral blood or in the lymph node. And it is trying to find and identify and measure these this reservoir that has caused problems with cure. Once you stop therapy, then these cells can activate and the variants can activate and therefore cause viral rebound and that thus cause disease. Next slide, please. Next slide, please. So HIV also has a large amount of viral diversity. So we have HIV-1 and HIV-2 and a huge amount of diversity. And it's been said that as compared to COVID and SARS-CoV-2, all the viral diversity in the entire world pales in comparison to the viral diversity of HIV in one patient that has HIV. So one thing to note here is not just the phylogeny, but I also want to point out specifically on this diagram, the SIV, which stands for simian immunodeficiency virus. HIV was thought to originate from simians and had a zoonotic transmission from simians over to humans. What in most simians, then SIV replicates but doesn't cause any disease, particularly any immunodeficiency, except in the rhesus macaque. The rhesus macaque has an immunodeficiency from SIV that's very similar to the immunodeficiency in humans. And thus, the rhesus macaque has been the long-standing animal model for the past few decades and has really helped us in the understanding of HIV, as well as therapies and advances. Next slide, please. So to understand where MPS may fit, we need to contrast how they can help, how they can complement the work in the animal models. And I think this group already knows plenty about that. But we're interested because MPS can better represent human physiology. Unlike other cell lines, many of these MPS can actually be infected with HIV, which enables studies of pathogenesis, host-pathogen interactions, and studies of the reservoir. We hope that as we integrate more immune cells and lymphoid tissues into MPS, that they can facilitate the study of HIV immune responses, which are a key barrier to further advances in the HIV research agenda. Also, application of MPS can help us in the therapeutic development in pharmacology, in silico modeling, pharmacodynamics, and safety. And in particular, such as in my branch, we think that the tissue sources and modifications can permit study of unique conditions, including co-infections and special populations, including maternal and pediatric populations. Next slide, please. So we think that there are a number of tissue sources and MPS models that could contribute to HIV research, and we've highlighted some here. I'm not going to go through the details, but in general, what's listed in the table on the right progresses cranially to caudally, and I'll highlight a few further down. But overall, we think that these are helpful in understanding a lot about HIV pathogenesis, the reservoir, inflammation, hopefully more about immune responses, but also about therapeutics and antibody development. And so let's go into some more details on the next slide, please. So brain MPS, particularly organoids, are one area that's been a focus of the HIV field. and it's been a more recent focus, and they've been used to study HIV neuropathogenesis, central nervous system latency, as well as HIV silencing. So if we look, we can understand that there are microglia, which are very important in these brain organoids. But organoids can often be created with neurons, astrocytes, microglia, and the microglia are an important component for understanding HIV pathogenesis. So the microglia can get infected by HIV, and you can get some lately infected microglia. But then when you have pro-inflammatory activators, then they can be productively infected, and you can get replication. And the replicating virus can lead to, of course, inflammation, as demonstrated here in these pathways, and lead to a lot of neurotoxicity, neurocognitive problems. And in children, you can have neurodevelopmental problems. Prior slide, please. What's also understood is that now although other cell types, particularly lymphoid tissues, are the main tissue sources for the HIV reservoir, the CNS is an important and prominent HIV reservoir. Also, what's starting to be understood are aspects of signaling and HIV repression and induction. So some things that have been discovered, such as our pathways, such as Neur1, which can actually help, as it's expressed, can recruit other factors that can silence HIV replication. Further, some are studying substance use and dependency, and so methamphetamines and opiates have also been shown to relate to HIV latency, induction, and silencing. Next slide, please. But the key tissues that we have are also our lymphoid tissues, as HIV particularly interacts with the immune system. So we have lymph nodes, and we have tonsils that are being used, although other tissues would be important, gut-associated lymphoid tissues, because gut are an important reservoir. But the germinal centers are important HIV sanctuary sites, where it's difficult to really control HIV there, and they can often hide from the immune system. But you can also see that tissues are available, some from tonsils on the right with macrophages, and we have interest in what we can do with these immune responses. Next slide, please. So there are certain key immune events that can be recapitulated in current and nascent models of lymphoid tissues. So lymph node MPS can maintain intact spatial structure. They can retain intricate chemokine gradients, respond ex vivo antigen challenges, and they can model some immune responses. We can study things like chemotaxis, motility, cell-cell communication, and perhaps organ-level function. And even more recently, there's been some work on adaptive immunity in response to live attenuated influenza vaccines. So you can see on the bottom right some responses in some tonsil germinal centers to LAIV as opposed to those that are not, and some additional antibody responses have been demonstrated. And the microfluidics may actually also allow vaccine drainage from injection sites and infector cell traffic to target organs for further studies of vaccines, which we would hope we can understand more about. Next slide, please. So some of the key questions we have now really relate to cure, vaccine, viral control, and we wonder, as immune responses and immune MPS and lymphoid MPS get further developed, can they contribute more to helping us understand these next frontiers and bottlenecks in HIV research? So we've had exciting news that has been over the past number of years that it's been hard to translate into clinical benefit for the larger population of HIV, a large HIV population. So we've had some stem cell transplants. The Berlin patient, which was the first one cured, he had acute myeloid leukemia, had chemotherapy, radiation, and underwent allogeneic hematopoietic stem cell transplant with stem cells that were CCR5 deficient had deletions at locus 32, which made them essentially resistant to HIV infection. The first woman undergoing cure with this mechanism was recently reported in Cell this past year. But we've also understood that early initiation of antiretroviral therapy in infants, such as one in the Mississippi baby at 30 hours of age, helped restrict reservoir formation, actually led to about 1.5 years of remission off of antiretroviral therapy. We also understand that some patients are able to spontaneously control HIV, and we're studying different viral reservoir characteristics and immune characteristics in that population. But we've also been working for decades on trying to get a vaccine, as in the bottom left. But the most recent trial of the Mosaico trial, as you can see, have very overlapping vaccine and placebo infections there. So can we understand more about why the immune system is insufficient to control or eradicate HIV? Why are we having all these unsuccessful HIV vaccine trials that showed promise and efficacy in animal models? And can we understand more about the combination cure strategies, such as shock and kill, where you try to give some latency reversal agent that allows the immune system to sense the infected cells and then amplify the immune response to kill off those cells, blocking and locking? So can you simply lock the integrated DNA so they never actually reactivate? And can we use gene therapies such as CRISPR-Cas9 to excise HIV, or can we use CAR T cells, vector-delivered broadly nucleoside antibodies? There are ways to take lessons from allogeneic hematopoietic stem cell transplants. So those combination strategies may be difficult outside of a full animal model, but perhaps components can be tested, such as using latency reversal agents, which may have safety concerns due to immune activation, but people have had difficulty showing how well these latency reversal agents actually decrease the reservoir in human tissues. Next slide, please. So one of the major complications of HIV is tuberculosis. Indeed, in people living with HIV, tuberculosis is the leading cause of mortality, and people with HIV have 18 times the risk of TB infection as opposed to people without HIV. But TB has interesting host immune responses. So initially you will inhale the bacterium, and then in the alveoli you might have initial macrophage responses and ultimately recruitment of T cells and B cells to form a granuloma. But then if the granuloma cannot adequately contain the infection, then you can get rupture and spread of the bacilli, causing more disease and causing TB disease. Now, lung-on-a-chip models have been developed and are being studied in TB. You have some that have here, such as on the figure on the right, component D, which with an alveolar liquid interface with vascular perfusion. And this one was developed in order to look at surfactant, because surfactant may provide defenses. If you look at the bottom left, well, it might be hard. I guess I don't have a pointer, but the macrophage, the surfactant, those with surfactant can better contain bacilli replication in the bottom right than those with deficient surfactant, with more overgrowth of bacteria on the bottom right. Next slide, please. Oh, sorry. Next slide. So I'm not seeing it move forward here, but if you are, let me go on to the.
I'm not seeing it either, Dwight, sorry. Is there a problem with the slides, Amber or John? Is it advancing on your end?
I am also not seeing. Oh, there it goes.
Okay, we go.
All right. Let me see if I can go quickly through last components. But we also think there's a lot of interest in there. MPS certainly were developed in a lot of ways for therapeutic evaluation. So we think that there may be particular applications of people living with HIV because they have combination antiretroviral therapy and combination and toxicities from the combinations, such as mitochondrial toxicity. And so some MPS are being studied, used to study mitochondrial toxicity from antiretrovirals. There are co-infections such as tuberculosis, which also require additional combination therapy. They can have organ dysfunction, as mentioned, and there's special situations such as pregnancy lactating women and pediatrics. And so a lot of these MPS can help with preclinical development, and especially when paired with in silico models of PBPK modeling, can help us with drug-drug interactions, compounding toxicities, including those that require have immune components such as drug-induced liver injury. And we hope that these can be adapted to special populations. Let's given time. Let's go to the next slide. In the maternal adolescent and pediatric research branch, we have special interest in the pregnant and pediatric populations because they have generally had delays or not had labeling for many of the drugs. In fact, if you look at antiretrovirals in pregnancy, in general, the median knowledge gap from FDA labeling to the first published PK data in pregnancy is typically 6 years. Next slide, please. So we think that placental models, which were actually mentioned in that Zika virus talk yesterday, can be helpful in regard first in understanding human pathogen interactions and immune responses, but also helping us understand transplacement transit of broadly neutralizing antibodies or small molecules. Next slide, please. Particularly important might also be teratogenicity evaluation. In 2018, there was a signal that dolutegravir may lead to neural tube defects. But it took years for us to actually follow and see that the effect is small and now no longer statistically significant, but it led to delays and probably increased transmission and increased maternal mortality by delaying dolutegravir rollout for this concern. Recently developed has been a human neural tube MPS, which, as you can see on the left, if you give is a neural tube that would typically have apical constriction under normal circumstances, but when given with, say, a known teratogen such as valproic acid in the bottom right, you fail to get apical constriction, you'd have a neural tube defect, which happens with valproic acid, but not for its non-teratogenic metabolite VCD. Next slide, please. And overall, we also think not just small molecules, but broadly neutralizing antibodies have importance for prevention and potentially ART-free treatment and cure. Next slide. And so in this slide, we see that some preliminary efficacy hopes are present, as in a study of prevention of horizontal HIV transmission using some broadly neutralizing antibody had about 76% efficacy for susceptible virions, but did not have much efficacy for those that were not. And then in another combination BNAB study in people living with HIV, the combination BNAB, the longer it was maintained, led to longer viral suppression and also some reservoir reduction and composition changes. Next slide. And so we, of course, have some outstanding questions of how these can still be applied and developed, further applied to the HIV research agenda, but we have hope for the MPS in this field. And next slide, please. And so thank you very much. I want to thank all the people who have contributed in our DATES working group. Thank you very much.
Thank you very much, Dwight. That's an excellent overview of the MPS applications in HIV. I think there is a question in the chat, which would be fantastic if you can get. I think in the interest of time, I would like to move on to the next speaker, if that's okay. But please do respond to that question in the chat, Dwight. And anybody else with further questions for Dwight, please do put them in the chat for him. Thank you very much, Dwight. Okay, so our next speaker is Landis Lopez Quezada. Landis, are you there?
Yes, I am. Can you hear me?
Fantastic. Yes, we can hear you and we can see you. So over to you.
So as Anthony pointed out, my name is Landis Lopez Quesada. I am a principal scientist here at Draper, and first of all, I did want to thank the organizers for giving us the opportunity to not only speak of our Predict96 system and how we have been able to model SARS-CoV-2, but also have tested the FDA-approved compounds in our system and shown efficacy. So I kind of wanted to start off, next slide, please. Talking about some of the challenges that the pandemic has presented translational scientists. And the efforts to find effective therapeutics against SARS-CoV-2 have recently been in this review article, they have been beautifully summarized, I should say, in this article, in which they spoke of, again, the challenges and also the achievements that that translational scientists and been able to achieve, because of the fact that basically, under normal circumstances in the past, we have been able to get therapeutic treatment for disease in a time scale that is measured in, let's say, a decade. For example, HIV that we just heard about, that took about a decade to get effective treatment in market. But with the SARS-CoV-2 pandemic, that had been accelerated down to maybe 2 years, which is a pretty big achievement for the field. But one thing that really struck out as I was reading through this is the fact that the most effective treatment that have been approved for use in the clinic relied greatly on the work on previous program and from previous outbreaks from different viruses, including MERS and SARS-CoV-1. So next slide, please. So it kind of highlighted the fact that where MPS really can shine, and it's in the novel therapeutics. So in reducing the number of false positives that are taken into clinical trials. So a great deal of time and resources are used in drugs that have shown efficacy in a 2D cell model or shown efficacy in animal models, but once taken into the clinic, they really are not as effective against or efficacious against patients. So I think this is an area where MPS, as we have heard from multiple other talks, can really shine. It can really decrease the number of compounds that are taken into clinical trials that are in the end not effective. Next slide. So at Draper, we have developed this fit-to-use tissue model, the Predict 96 system, as a call for use as one of these tools that can accelerate the drug discovery pipeline. So next slide. The Predict 96 system is mainly 3 components. So unfortunately, I don't have a pointer, but is in the control module, which will control the pneumatic pumps that feed the perfusion of the tissue. So again, and then followed by the lid, the sterilized lid, which sits over the culture plate. Now this lid has 192 micro pumps that are integrated into the culture plate, and that will allow the fluid flow both at the apical and in the basal side. Now, in particular for our lung model, so as you can see here on the tissue plate, since we maintain the mature tissue at the air-liquid interface, we really only use the pump system in the basal ports. And again, so the cells will sit on these micropores membrane, followed by a steady perfusion throughout the maturation process in through the basal port. Next slide, please. So for the tissue itself, we are able to use primary human cells. So these cells can either come directly from patients who suffer from different comorbidities or from commercial tissue sources. The mature tissue itself is approximately 40 to 50 microns thick. And as you can see from the images on the lower half of the slide, when you look at the apical, looking from a top-down view, you could see that after maturation, you have representation of all the major cell types in our tracheobronchial airway model. Again, so you have your basal cells, goblet cells, and of course, your ciliated cells. Looking at a cross-section, so next slide. So this is, of course, the adapted Predict 96 ALI model that we will be using for our infection. So next slide. And of course, looking at a cross section of the human lung biopsy, you could see again that you have this beautiful pseudo structure that mirrors very well the human lung. So you have this pseudostratification of your cells where you have your ciliated cells that can, and you have a recapitulation of the mucociliary movement on the ALI. So again, the Predict 96 system has this very high throughput because all these independently operated 96 tissues can again be operated independently of each other. So you're presenting again a high throughput, which is very, very important when you go there to screen lead compounds for their efficacy. Next slide. So when we were considering, we were using this model for SARS-CoV-2, one of the first questions we asked is this tissue permissive for infection? So as you could see here, again, this is data from 2 separate donors in which you could see that you have expression of the 2 major mediators of viral entry, the ACE2 receptor and the TMPRSS2 receptor. So we get really good expression of both of these mediators of viral entry into a tissue. Next slide. And indeed, we were able to get a very robust. So this data here is looking at infection of our lung model with the Washington strain, and we get very robust infection throughout a 6-day time course of again different donors of the tissue. Again, and you could see from the cross-sectional studies of the expression of the nucleocapsid protein on our tissue that even between donors, you do see some modest differentiation between the donors. So we're able to capture those differences that can exist between donors. And of course, from the top-down view, you could see a really good, robust viral replication and dissemination. Next slide. And this is something that we have been able to recapitulate with the different variants of interest, of concern. So we can see here that both Omicron, Delta, as well as previously showed, the Washington strain are able to robustly replicate in our model. But in addition to that, when you look at the differences between the replication rate and the infection kinetics of these different variants, the model is able to capture these minor differences that can exist. And again, these are just images of the robust infection of the model in different donors. Next slide. And I mean, this is just highlighting one particular Omicron, which of interest. So you could recapitulate again that infection across multiple tissue donor types. Next slide. So how we're able to infect our tissue while maintaining the lung tissue and ALI is essentially once we have matured the tissue in ALI with again a constant pumping throughout on the port channel, on the basal channel. When we go to infect, we do about a 2-hour infection, particularly low MOI. After that 2-hour infection, the virus is washed off. Any virus that has not adhered to or entered the cell is washed off. And then the tissue is quickly returned back into ALI. Now, one thing that I do want to highlight is when we do antiviral treatment of the infected tissue, this is all administered through the port channel at very low volume, and that is constantly changed every few days to kind of simulate a consistent, a continuous course of treatment of the tissue. At the end of our infection, we're then able to then either fix the tissue, and again, this is all occurring within the BL3 environment, and after that, we fix the tissue and then go on to do some microscopy. Next slide. So the multiplex assays that we're able to carry out in this model include, QRT-PCR, TCID50. We do follow up our RT-qPCR data with plaque assays to verify, again, either through TCID50 or plaque assays, our results. But again, these are 96 individual devices in which we can now carry out, even throughout the course of infection, different assays, including biomarker analysis like Luminex and ELISA. We have also carried out transcriptomics and proteomic analysis of the tissue that has been infected. Next slide, please. But very importantly, especially when you're doing kind of drug screening, is how reproducible is our data, is our infection, not just between all the different 96 devices, but also between different experiments. And we do get very high reproducibility across, again, from the device to device, as well as from plate to plate. Next slide. And I kind of want to come back again to that central question, which is our goal, especially here when we work with NPS system, is improving the timeline that we can get effective treatment into the clinics. And one way to do that, again, is to decrease resources that are used for compounds that may have worked in an animal model, but then once it reaches human, it is not effective. So we implemented so next slide. So we went and looked to see, can our model recapitulate what we see in patients? So we screened the FDA-approved compounds in our infection model to see how effective are they. And we did see a recapitulation of the effects that we see in the clinics. For example, nirmatrelvir, remdesivir were very effective at reducing viral loads across our 6-day infection. And because, again, of the level of throughput that we're able to achieve in these compounds, not only can we follow their effect on viral kinetics through a multiple days during the course of infection, but at multiple concentrations. And next slide, please. We also were able to use microscopy to kind of verify what we're seeing with the QRT-PCR data. And this, of course, is showing you that, just highlight a particular case of nirmatrelvir, where we see that the percent of nucleocapsid positive cells were significantly reduced as you increase the concentration of treatment. So this is the day 6 data. Next slide, please. And again, as I said, one of the key things is can our NPS system be predictive of what we would expect to see in the clinic should these compounds be brought into clinical trials. And we did see that our most active compounds did, or at least in this particular patient cohort, did mirror, again, what we saw in terms of the reduction in the risk of hospitalization rate, where remdesivir and nirmatrelvir had very good activity. And also in the clinic, we also see that these are most effective also in these particular patient cohorts. So next slide, please. So I guess in summary, in terms of using the Predict 96 system, we were able to show, again, that that the system can be used to kind of predict potentially how effective a compound will be once it reaches clinical trials. So I do want to highlight this as a therapeutic because of its throughput, that can be used as a therapeutic screening platform for your lead compounds, either completely novel compounds or repurposed compounds before they reach while they're still in preclinical stage. And although I demonstrated this model for SARS-CoV-2, it is adaptable not only to the BSL-3, but also we have adapted into the BSL-4 environment. And again, this is a proof of concept study that can show that we can match the clinical outcomes. And we think that this can accelerate that drug development pipeline down the line. So I do, I just got the warning, I do want to highlight that although I have shown you the lung model for the Predict96 system, if you don't mind going to the next slide. At Draper, we have adapted this system to multiple other organs, which can expand not only to infectious disease modeling, but also toxicology. Would you mind going back? So this has been shown to be a very good system to model different organ tissue, including blood-brain barrier, liver, kidney, as well as a vascular model. Again, highly amendable and high throughput for use across different uses. Next slide. And with that, I do want to thank not only our team, but also our collaborators at Mass General and Tufts, where we do most of our BL3 work. And I'm open. I don't think we have a lot of time for questions, but if there are, please.
Thank you, Landis. A lovely presentation. You answered my question for me in terms of the applicability of the platform in other organ systems. So great to see it's as widely applicable as it is. I'm not seeing any questions at the current time in the chat, but obviously if you'll be around for the rest of the meeting. So if anybody has questions for you, please do include them within the chat, and Landis will address those directly. Thank you very much, Landis. Okay, so now our last presentation before we have a break is from Sayum Ahuni. Are you there, Sayum? I can see your slides, but I'm not hearing you.
Can you hear me?
Yes, we can hear you now and we can see you. Fantastic.
Okay.
So, over to you. Thank you, Sayum.
Thank you, Tony. I am Sayum Ahuni, and I am I'm leading the R&D group here at MatTech Corporation, which is now a Bico company. And I'm a virologist by training, but later get into tissue engineering space. MatTech is, as you all know, MatTech is one of the oldest tissue engineering companies that started producing organotypic tissue models since 1993. And through the years, we have validated models like the skin and ocular tissues for irritation studies, and also other organ models like the epivaginal tissue, which is now under consideration for validation for feminine to study irritation potential of feminine care products by FDA. So we are a company now. We had two facilities, one in Ashland and another one in Slovakia for the European to serve the European researchers, and also we have distributor in Japan, Corabo. So our tissues can be shipped globally, globally to different regions. So as you all know, drug development process utilizes animal animals for preclinical testing. And these models are costly, time-consuming, and less predictive. Results from animal models are less translatable to human responses. So some of the interesting results that I'm going to show you will help us to understand how these models can be utilized for infectious diseases. Next slide, please. As you can see, this is a template for our technology for reconstructing tissues. What we do is we took the tissues, organ tissues from donors, and we isolate the cells, and then we isolate epithelial cells, fibroblasts, and endothelial cells, and so on. Then we expand them in culture, and we cryopreserve them. After we cryopreserve them, we take, we have 1 vial of sample, and then we can we seed them onto a tissue culture insert with a microporous membrane, and those microporous then we allow the tissue to grow under air-liquid interface, and that leads to tissue differentiation and stratification to give you a full differentiated tissue, in this case, like say the nasal, airway, alveolar, or intestinal tissue models. So MatTek has now developed over 15 tissue models from different organs, and some of them are widely used by the pharmaceutical industry. Next slide, please. So our technology looks like this. We have the insert, as you can see it from the left side. You have the insert, and we seed the cells in there, and under air liquid interface condition, we just differentiate the tissue. And this inserts the cell seeded onto a microporous membrane under air liquid interface, and then the formats could be 6-well plates or 24-well plates and 96-well plates. And these are some of the things that we produce day in and day out. They are well characterized. Our QC criteria is stringent, and because it has to be reproducible when you ship it outside of our labs. So it's used by different people, and now we have over 1,000 publications using our MatTek models. So the next slide, please. So as we can see, these models can be used independently, or they can be incorporated into microphysiological systems. And MatTek was one of the companies early on who was involved in developing the microphysiological systems, by collaborating with the MIT group for the DARPA project. Some of the models, like the small intestinal tissue, were developed from the DARPA project. So in there, the first thing that we were saying that we're coming, concept that was developing at that time was to use these models, to incorporate these models into a microphysiological system. So some of the microphysiological system or the chip platforms start to utilize inserting our tissue models or inserts into their format. So that's how we were basically involved starting from the very beginning. And as you can see here, we had a good partnership with tissues early on, and that partnership is still going. We were able to co-culture the liver and the intestine together, and in a microphysiological system. And what we have been looking there was we were able to develop media conditions that can support the liver tissue and the intestinal tissue together for culturing for up to 28 days in culture. So with the development of these models, I think the most important thing that we have to focus also give attention to is, do we have the right media compositions that can support different organ systems at the same time? that is a more complex issue because we don't have any universal media at this time. So those are type of things that we are involved in. When we develop models, we develop also the appropriate media composition and so on. So our intestinal tissues have been also, for example, used in multiple formats of the microphysiological systems. To my knowledge, there were about 4 microphysiological systems that have been using our format. So those are some of the contributions MatTek has done to the microphysiological systems. Next slide, please. So because this is the workshop is on the infectious disease, I will try to bring the utilization of these models for infectious disease, particularly for COVID-19 research. So as you can see, we have epinasal, airway, and alveolar tissue models that have been developed at MatTek and used for various research activities. So the epinasal tissue was developed at the time of SARS-CoV-2 infection as emerged when researchers identified nasal epithelium as a portal of entry for infection and transmission. Its development was first tracked to make it available to aid scientists in the fight against COVID-19. So since then, we have over 20 publications that use our tissue models for COVID-19 research. So MatTek's human respiratory tissues have been widely used starting from the very beginning. And in fact, some of the models, like the alveolar models, have been one of the first ones to show that a long infection of COVID-19 can be detected up to say up to 50 days after infection using alveolar tissue models. Next slide, please. So when we make tissue development, we give attention to the characterization to make sure that it mimics the in vivo situation, because it has to be more physiological for a more physiological response. So as you can see, this is the new epinasal tissue models that we have developed. And you can see there are, in figure 4, you can see there are goblet cell markers in red, and also the cilia markers in green on figure 3, and also the SE2, which is a co-receptor for COVID-19 on figure on the right side lower bottom figure. So we characterize the cells, and they have the right markers and the right structure to proceed to any type of utility. Next slide, please. We also collaborated with Jackson Lab to characterize this model at a single cell level. And what they did was they did a single cell characterization of the tissue, and what they found is that the different composition of the cells are there. In B and C, you can see the co-receptors for COVID-19 or SARS-CoV-2, and in this single cell analysis, they have identified previously identified cells, and these cells were enriched in response to allergic rhinitis virus, respiratory syncytial viral infection, and the increase in cytokine and metalloproteases expression, and some of the lamins and integrins. So infection of this virus showed also some markers that are associated with viral infection. Next slide, please. The airway model has been also characterized by Emily Lee, for example, at NIH. And as you can see, You have the APRY tissue, SE2, TMP-RSS2, NRP1, and alveolar also shows the same expression pattern. And those are key components for if we are going to proceed with COVID, SARS-CoV-2 infection. Next slide, please. So one of here is a schematic. So when we proceed with using these models for viral infection experiments, this is a schematic representation. You have the tissue, and because this is different from organoids, at the upper you have exposure for the you can expose the upper surface. So the apical surfaces you can add viruses or chemicals or drugs in there, and then you can add drugs to the apical surface or the lateral surface, depending on where that response is going to be expected. For example, you can for orally administered drugs you can add to the surface, but for systemic drugs you can add it into the culture supernatant, and then you look for infection in up to 6 days. And after the infection, you can harvest apical washes at different time points, and then you can determine viral titers. Next slide, please. And this is an example for airway and epinasal tissue model infected with the wild type or the Washington type, the beta, delta, and Omicron viruses. So as you can, this was in collaboration with Utah University, University of Utah. They did these experiments, and when they did it, you see the viral peak replication time was about 5 days in the epi airway, and the epinasal was about 4 days. So you can see these viral replications in these systems. Once you establish this viral replication capacity, then we were able to get into drug responses. Next slide, please. So as you can see, these are 2 drugs that has been tested, and as you can see a dose response for remdesivir and also AIDD 1931. So these are some of the examples that you can use these models for viral infection and to see drug efficacy also. Next slide, please. And the antiviral activity of M128533 also was monitored on this tissue model, and as you can see that there are different responses for in the airway tissue model. So this one, the next one I'm going to take you is for small intestinal tissue model. Most of the infection experiments has been done on airway tissue, but the neglected one was intestinal tissue, which is probably maybe one of the reservoir for COVID-19 viruses. So you can grow this tissue, as you can see it, in a polycarbonate membrane or on a PLJ membrane, which is biodegradable, and you can incorporate other cell types like fibroblasts and immune cells into this system. Next slide, please. And this was one of the applications from Robert Davis group here at Boston University that we collaborate with, and we're trying to see they were screening repurposed drugs, about 6,710 FDA approved drugs, and from there they narrowed down the most efficient ones, and they did infect the small intestinal tissue model with SARS-CoV-2, and they tested the different drug candidates. And as you can see here, obaclac's, that was a cancer drug, that was one of the most efficient drugs to inhibit HIV, inhibit SARS-CoV-2 infection in the intestinal tissue model. So these models can be used for pathogen host interaction studies and also for drug efficacy studies. The intestinal model, which we have been published earlier also with Mass General Hospital, we have been studying the mechanism of Shigella viruses in the small intestinal tissue model. So those are type of applications that you can use these models. Next slide, please. Then with a request from a company, we were trying to incorporate immune cells into the system. And you can see we are able to incorporate immune cells. And as the tissue differentiated, you can see macrophages inside this tissue model. And that was a big success for us. And we tried to see if, in fact, this incorporation of immune cells can have can exacerbate immune responses. Next slide, please. So as you can see, the incorporation of immune cells in red, there you can see some of the cytokines like the chemokine cytokines like CCL20, RANTES, TNF-alpha, IL-8. Those were exacerbated upon the addition of intestinal Langerhans cells into the system, or dendritic cells into the system. So these are some of the things that we want to pursue. And in fact, we did with gene markers, with the collaboration with gene markers, we did microarray analysis for about 23,000 genes, and we have seen a lot of responses based on innate immune responses of this dendritic cell containing model. Next slide, please. To summarize it, these models have human relevance. These tissue models are derived from human cells and their primary cells, so their biology is compared to a human. There are tissue functionality, they are reproducible, and they are predictive, and they can be compatible. They have compatibility with microphysiological systems. Next slide. Thank you so much.
Thank you, Sayum, for a really wonderful presentation of how NPS can be applied to explore the response of SARS-CoV-2 across the different parts of the respiratory tract. So we do have a question in the chat from Jeffrey Borenstein. So great presentation, Sayum. As you and Landis showed, there are many key experiments that cannot be done with pseudoviruses in a BSL-2. Would you have expected that based on clinical trials, your Omicron viral replication would have risen more rapidly than the other variants?
Can you say it again?
So would you have expected that based on clinical results, your Omicron viral replication would have risen more rapidly than the other variants?
Looks like that, but you know, when you model this type of things, this is a single organ. You know, we have it separately, the nasal, the airway, and alveolar. But to get that conclusion, probably something, if you can simulate those three things, with you infect the nasal first as a first entry site, and then see where the virus is going on. And if you completely follow those, probably you can give more conclusion. But these are short experiments. I think they to get into that type of conclusion, probably we have to do more replicate experiments.
Thank you. Thank you very much. So there's no further questions, Sayum. So thank you. Obviously, if any questions come up into the chat, please do respond to them. I'd like to thank all of the speakers from that first session. A great series of talks that will lead us nicely into the remaining session after the break. So we're a little bit over time, but I'd be grateful if we could try and remain on track with the program, and if we could try and get back at about 35 minutes past the hour for the next session. Thank you all very much, and we'll see you soon. Welcome back, everybody. I hope you've all returned fully refreshed. for the remainder of this second day of the MPS Core Spring Workshop. So we have a couple of presentations before we get into the panel discussion, and the first of our presentations this afternoon is from Tanya Suligoj. Tanya, are you there?
I'm here. I'm here. Can you hear me?
Fantastic. Yes, we can hear you and we can see you. So over to you. Thank you.
Thank you very much. My name is Tanya Shuligor. I'm a postdoc in Natalie Juju's group at Quadrum Institute Bioscience. I'd like to thank the organizers for the opportunity to present our work. I will talk about my journey in MPS use and the steps and challenges that we experienced along the transfer of containment level 2 to containment level 3 setting. As you probably know, containment level 3 is the UK equivalent of BSL 3. Next slide, please. So let me start by a quick introduction. Quadrum is in the UK in Norwich, which is about 2 hours away from London and about 1 hour away from Cambridge. And in this new building, we have an endoscopy unit on the ground floor. Next slide, please. Our institute is a part of Norwich Research Park, which is a cluster of several institutions with lots of experience in microbiology and expertise. There's also a regional Norfolk and Norwich University Hospital, as well as the biorepository that facilitate our access to primary human tissue. Next slide, please. Our science is focused on gut microbes, food, and health, and we use interdisciplinary approaches and are also mindful of 3 Rs. Our institute invested in a range of advanced in vitro models of human gut to reduce our reliance on animals. The technology that we use for gut microbiota include various in vitro colon models, including the advanced model known as SHIME, which stands for the Simulator of Human Intestinal Microbial System. And for the host side of things, we have the MPS platform for gut on a chip. And my interest is in the gut, and we started using emulate technology back in 2019 after I spent 6 weeks training at Emulate in Boston. Next slide, please. Just to tell you about our workflow, we obtain fresh human samples downstairs in the endoscopy department, where we can access biopsies as well as matched fecal samples and bloods if we need them. And then we take these samples upstairs for processing. And in the case of biopsies, we first extract crypts, which we put in extracellular matrix proteins and define medium to generate organoids. These organoids are then used either in gut on chip or we can freeze them down and biobank them. And we have generated a biobank from all parts of colon, proximal transverse, and distal colon. So next slide, please. So for those who aren't so familiar with emulate gut on chip model, we use 2-channel silicon chips that are commercially available, which are separated with a porous membrane. And first we coat both parts of the membrane with extracellular matrix proteins, and then seed the cells. So endothelial cells in the bottom channel and gut organoids in the top channel. And then when the chips are ready, we then connect them to the pods. Pods are inserted to Zoes, and Zoes then run by the orb, which controls the microfluidic flow. And also in the case of gut on chip, also introduces peristalsis like stretch. And we can easily control the flow rates and the extent of the stretch. And it's very straightforward to administer any test substances. The good thing about this model is that we can very easily sample what's coming out of both channel over time. And this was the approach used in my paper. So as mentioned, we established the chips, gut on chip using emulate protocols. And in this study, we used them to test the effects of fermented human milk oligosaccharides on short HMOs on gut barrier function. We chose 2 human milk oligosaccharides, 2FL and LNNT, and have them fermented in vitro by Scheim, which was inoculated with human fecal microbiota. And then, so obviously using this advanced in vitro colon model. And then the resulting metabolites from this fermentation were then used to treat gut-on-chip with a view to analyze cytokine profile as well as gene expression. We carried out this experiment with human organoids from proximal, transverse, and distal colon model, distal colon from from 3 different adult donors. And this is just on the right-hand side. This is just a snapshot of one of the results where we show that barrier function gene Claudin 5, which is circulated, is upregulated upon treatment with fermented 2FL. And then the results of this study then supported the use of HMOs as a potential food supplement to strengthen the gut barrier function in adults. Next slide, please. We have used this model to support several projects across our institute to study different aspects of gut health. For example, similar approach that I've just described was also used for another foodstuff, namely fermented resistant starch. And we also looked at the effect of fermented material on adult gut barrier function. We further use gut-on-chip for co-cultures with bacteria, for example, pathogens like Campylobacter jejuni, as well as commensal bacteria like Ruminococcus gnavus. And this technology is well suited also to study the fate within the epithelium for outer membrane vesicles, as well as bacteriophages, which is something that I'm currently working on. And we are always interested in advancing the model through the close engagement with Emulate by taking part in their beta test, we contributed to the development of advanced models, including immune cells and disrupting gut barrier function to study leaky gut. Next slide, please. And then COVID came. Given the expertise in the gut-on-chip model, we were approached by UKHSA to help set up the models for human lung with the view to use them in infectious disease research. Emulate technology is a versatile platform which enabled us to establish both airway and alveolus models in relatively short time. Next slide, please. So in airway model, we use commercially available human small airway epithelial cells isolated from bronchiole area, which is seated in top epithelial, which is epithelial channel. The cells are cultured in air liquid interface, which means that there's no medium above them, but just air. And in this way, we mimic in vitro conditions that is close to what occurs in vivo. Air liquid interface also supports the cell differentiation, and using a phase contrast microscopy, one can observe a liquid interface with this dark band that we see there on the left and right, but it's for the airway chips, yeah. And we have done quality control on these chips, looking into apparent permeability, which we often refer to as PAP, and that one was also in physiologically relevant range. PAP is evaluated by measuring the amount of fluorescence probe that passes from basolateral compartment of the chip to the apical. So the greater the amount, the more leaky is the barrier. And then for this experiment, it typically takes a few hours to complete. So we need to submerge air liquid interface to liquid-liquid interface, but after the end of the experiment, we then reestablish air liquid interface. And then as you can see on the right, we also established alveolus on chip, which uses 2 cell types, primary human alveolar epithelial cells, as well as human lung microvascular endothelial cells. Epithelial cells are also cultured in air liquid interface, which supports barrier function of the epithelium. And when the cells are ready, we introduced physiologically relevant stretch that mimics breathing-like motions. And we have also done QC with PAP assessment on these chips. Next slides, please. We characterized the chips with immunofluorescence and confirmed the presence of relevant cell types. In the airway, we stained for beta tubulin to show the presence of ciliated cells, CC10 for basal cells, CK5 for club cells, MUC5AC for goblet cells. Can you press one forward, please? Yeah, and here we also show the cilia beating, which basically sweep mucus and any other dirt of the lungs. And then on the right-hand side, we see that we demonstrated that in our alveolus chips, we do have type 2 as well as type 1 cells present. Next slide, please. So next, the lessons learned from these models were then used for application to infectious disease research. We ran our first infection experiment with the flu virus. This was Perth isolate of flu virus H3N2, which was provided by UK HSA. And we've used Amulet protocol as a basis for this work that I'm going to show here. So we used human primary alveolar epithelial cells and lung endothelial cells to see the chips. And after connecting them to the pods and Zoe, we introduced air-liquid interface and performed quality control experiments by assessing PAP. And then we inoculated the chips on day 7 of air-liquid interface by flooding the air-liquid interface. But just to point out that this experiment was primarily aimed to just set up the flow workflow for this virus infection and the desired downstream analysis. So after the inoculation, we re-established a liquid interface, and then over 3 days, we were then harvesting the media, the basolateral effluents that came out of the chip. And this was at 6, 24, 48, and 72 hours post-infection. We also were acquiring phase contrast images and collected washouts from both chip channels to assess the titers of viable flu virus with focus foaming assay and fix the chips for subsequent immunofluorescence analysis. Next slide, please. And just very briefly, brief comment on the results. We observed the infection by phase contrast microscopy. However, the cytokine response was very low overall. Next slide, please. But really what we were aiming for is just basically how we wanted to adapt the workflow to perform the alveolus and chip experiment using authentic SARS-CoV-2 in CL3 setting. Next slide, please. We used similar experimental design that I've just shown, apart from the fact that we also introduced stretch that mimic breathing-like motion. To tell you about SARS-CoV-2 virus, this was actually VIC-1 that was isolated from Wuhan-acquired case of a man who returned to Victoria second half of January 2020. And this virus was also provided by UK HSA. Next slide, please. And here I'm just summarizing the adaptations that we needed to take, the infection experiment from CL2 to CL3 setting. In terms of equipment use, we needed a separate kit to be placed in CL3 lab in addition to the existing CL2 kit. And we really thought about how to minimize the number of chip manipulations. And that one of them was, for example, that we used the zoestat and ORP to actually inoculate the chips by the flow rather than doing them by hand. We also, in terms of other manipulation, when it comes to reestablishing a liquid interface, as well as sampling and washes, in both cases, we reduced the flow rates, but prolonged the time just to kind of allow everything to really be collected or removed, but really also reducing the shear stress on the cells. And as we know, not everything can be done then for downstream analysis in CL2. So we had to deactivate the virus using appropriate methods that are compatible for downstream work. In the case for confocal microscopy, we use 4% PFA. And for instance, for cytokine analysis, we were adding Triton to deactivate the virus in the media. And some of these a part of this work, this was a collaborative effort with UKHSA. So we were sending live viruses that were basically the washouts from the chips to UKHSA for subsequent analysis. And in this case, of course, we had to make sure that everything is decontaminated and the samples are triple contained, had to follow appropriate shipping, courier service, etc. Next slide, please. And there's just a couple of figures to show you the results of our experiment done at Quadrum on alveolus chip with SARS-CoV-2 experiment, with SARS-CoV-2 virus, sorry. And this is unpublished data, which we aim to submit to the journal in the coming weeks. But what we see here is the confocal images of mock and SARS-CoV-2 treated alveolus chips 72 hours post-infection. Alveolar type 2 cells, which are targeted by the virus, are shown in red, cell nuclei in blue, and titration occludin in green. And we see that occludin is disrupted in SARS-CoV-2 treated chips, whereas in mock treated chips, the nice pigment-like structure is maintained. And on the right-hand side, we are looking at the figure showing the results of analysis of basolateral effluents from the chips, that we used to assess host immune response to SARS-CoV-2 over the course of 3 days. And several cytokines, the chemokines and ligands were tested with multiplex assay, but I'm also showing just a selection of these. Just to mention, the blue ones are 6-hour, violet is 24, pink is 48, and green is 72-hour time point samples. And what we can very clearly see that the differences between mock and SARS-CoV-2 treated chips are really become evident after 48 hours post-inoculation and later. So next slide, please. So in conclusion, I hope I managed to show that MPS platform is versatile and it enables several applications like what we've shown for our gut-on-chip work. And for us, it was relatively easy to switch from gut-on-chip to lung-on-chip. And we can relatively easy use the platform both in CL2 and CL3 setting. I have shown that alveolus on-chip model is suitable for studying SARS-CoV-2 infection. And I'm also basically showing that there is a hidden capacity, perhaps around the world with institutes like ours, where we do have MPS platforms, as well as CL3 access to contribute to the global effort to fight infectious disease. Next slide, please. And with this, I would like to thank my supervisor, Natalie, and other staff, employees at Quadrum Institute, particularly Kath from Bioimaging Facility and CL3 support, Gemma and Sandra, as well as Health and Safety, Rachel and Samantha. I would need to acknowledge that the lung part of the work was supported by FDA, and I need to acknowledge UKHSA, Naomi, Kevin and Simon, as well as continued support by Emulate, Sasha, Nasia and engineers, and thank you for your attention.
Thank you very much, Tanya. Wonderful presentation. So we do have a clarifying question in the chat from Sushma Bosel. Do you seed both epithelial and endothelial cells on the same day for the alveolus chips?
No. Endothelial cells are seeded later.
Okay, thank you.
I think 3 days later. Yeah.
3 days.
I think so, yeah.
And then in terms of the, this might be a naive question, in terms of the CL3 facilities that you've got, is that a facility that other researchers can access and use, or is it purely for Quadrum research? I mean, I don't know myself how widely available those facilities are within the UK.
We usually to be able to access it, we have to follow local rules, risk assessment, SOPs, etc. But it does involve quite intense training, and whether we can allow external people in, it would really very much depend on their qualifications, I think. But I'd rather not say too much because I don't
You don't want a sudden influx of
like, oh my
researchers.
God. No, but we have generally we try to to support collaborations in all aspects we possibly can.
Fantastic, excellent. Okay, thank you very much, Tanya.
Thank you.
And so that takes us very nicely to the last presentation before we enter the panel discussion. And this is from Simon Funnell from the UK Health Security Agency.
Thank you, Anthony, and thank you to the organisers for inviting me to speak today. As you can see from the slide, my name is Simon Funnell. I'm a scientific leader at UKHSA. Can I have the next slide, please? And then could you just move that arrow sideways? It's in the middle of the screen as well, please. Thanks. I'm a UKHSA employee. I also have a part-time place at the Quantum Institute, and I also work for the WHO. I don't have any patents to declare. Next slide, please. So this presentation will consist of 4 elements. I'll give you a brief profile of UKHSA for the people online. I know there's 60 people watching. Some of them may not know about UKHSA. I'm going to talk about some preliminary ORF1A in vivo findings, which I think is very relevant to a discussion that we'll have later on. I want to talk about MPS development at UKHSA, what we're doing, and also, as requested by the organisers, my perceived gaps and needs in this area. Next slide, please. So regarding the UKHSA, we have a wide range of analytical services that we can apply to each of these 7 model areas, and starting with insects, cell culture, small animal models, and non-human primate models of infection. But ultimately, we also conduct or participate in human clinical trials of pediatric vaccines and other types of human trials. And this is all designed to assist in the translational research required to license both vaccine therapeutics and drugs. And we do this within a framework of regulatory compliance, both from quality, and we can go up to GLP, and biosafety and biosecurity, we can get up to BSL 4 or a category level 4. And we also have tight ethical restraints, and we also comply to international regulations as when required. Oh, next slide, please. So this is just an overview of 15 to 20 years worth of research that I've personally been involved in at UKHSA over the years. The red lines indicate the organisms that we've utilized for in vitro MIC antibiotic assessment. The orange lines indicate small animal models of infection for the organisms listed that have been set up for the development, for the use of evaluations of antibiotics, vaccines, and therapeutics. And the green lines indicate models of infections that were set up in non-human primates for the use of and evaluation of antibiotics, vaccines, and therapeutics as well. As you can see, that's a broad portfolio, and it's 3-dimensional, being in each of the different models. And this list is by no means exhaustive of all the organisms that UKHSA has worked with, and it shows how adaptable and agile we are in addressing infectious disease threats as they emerge. And I've got to say that there are insect models available on site as well. Next slide, please. So I'm going to talk about an FDA and NIH initiative, which has been set up and funded through the University of Liverpool to UKHSA and a number of other entities, of which Quadrum and University of Hull are also participants. Next slide, please. More recently, that funding stream has been supplemented by another contract from the Wyss Institute directly to the UKHSA to develop a non-human primate organ on chip. Next slide, please. In addition, I'm involved in a really important project called the Agility Project, which has been funded by CEPI. This project was part of the COVAX Enabling Science SWAT team, And we were basically set up to look at the GISAID data streams, pick out variants of concern, look and see how they replicate, and assess their resistance to neutralization in neutralization tests. And if that was found to be interesting, we could take it forward into a hamster model for in vivo assessment of virulence. Next slide, please. During the Agility project, we have generated an enormous amount of data, and this slide summarizes several years of research that a very large team of people have generated and contributed to. Now, the brown box and whisper plots indicate neutralization activity of convalescent sera from an early prototype infection. And you can see as we go across that spectrum of SARS-CoV-2 isolates, that original convalescent sera has lost its ability to neutralize the Omicron variants. In parallel, the WHO standard 21136 has also lost its ability to neutralize Omicron and its variants. The green triangle also shows that NIBSC standard 234, although it looked like it was promising for Omicron, it lost its ability to neutralize BA4 and BA5, who perform very similarly for both the convalescent sera and for that working standard. The working standard 338, which is shown in the blue circles, has thankfully reacted very well with all of the Omicron variants that we've tested to date. But it's sort of interesting, before I move on to the next slide, to just look at the difference between BA4 and BA5, and basically there isn't much of a difference there at all. So next slide, please. However, when we put BA4 and BA5 into hamsters for virulence assessments, we were shocked at the difference between them. Now, this title says unpublished data, and at the time that I put the slide together, it was, but in fact, now this paper has been published by my colleagues, Davies et al., and you can, the DOI is presented there in the bottom left-hand corner. Basically, what you see is in BA5, you see a large reduction in weight at about day 7, whereas in BA4, we don't see a weight reduction at all in hamsters. And this is highly reproducible across the group, as identified in the stats on the right-hand side, which summarize the peak weight loss from those two groups of BA4 and BA5 infected hamsters. Next slide, please. And this isn't just because BA4 hasn't replicated. As you can see in the top right-hand corner, the virus is actually replicating in the upper respiratory tract of both BA4 and BA5 infected animals. And admittedly, there is more virus detected at day 8 in the BA5 infected animals. But in panel B, you can see that there's a small but significant difference in the lung at day 7 between BA4 and BA5 animals, but there's plenty of virus there, 10 to the 6 and 10 to the 7 log copies per gram. And in the last panel C, there's actually no difference at all in the viral load at day 28 left in both sets of hamsters infected with BA4 and BA5. Next slide, please. But the most shocking outcome was that BA4 infected hamsters showed virtually no clinical signs of infection whatsoever. There was only a small show of labored breathing at day halfway through day 4. But in the BA5 infected hamsters, we had this whole rainbow of clinical signs, which included ruffled fur, wasp wasted, a little bit of dehydration, and a lot of labored breathing in virtually all of the animals. And this was quite a remarkable difference, given that there was very similar levels of virus detected by PCR in both sets of animals. Now, why this is so surprising is that the BA4 and BA5 have basically got an identical spike. And so this difference in pathogenicity cannot be attributed to a spike change. And it's more likely that this difference is attributable to a deletion of 3 amino acids in the ORF1 gene of SARS-CoV-2, which is present in BA4 and not present in BA5. And I think this is a clue, yes, another clue to the function of ORF1. And this is not an isolated finding. Other people have found very similar things in other studies. And this is where human microphysiological systems will immediately be able to assist us, because if we can assess whether this is clinically significant by looking at human MPS in the way that Tani has just beautifully demonstrated. We could see if there are differences in cytokine induction in human cultures that are infected with BA4 and BA5. Next slide, please. A couple of days ago, I heard in an Emerging WHO ECDC meeting a story about sotrovimab, which has very low activity in a neutralization assay using Vera ACE2 tempra cells. But when you take that therapeutic and use it in hamsters and humans, it does actually work very well. So there's this gaping difference between the clinical utility of this drug and its use in a neutralization assay using Vero cells. So this is yet another area where MPS might help to dissect what's going on here. Is this just about the FC portion of that antibody therapeutic being critical? Do we need an immune element? Or is there a specific human interaction that's involved here? Or are there some cellular immunity effects? Or is the specificity of the monoclonal not entirely all about spike? We don't know that because the Vera ACE2 Temperus assay is spike focused, unlike the MPS assays. Next slide, please. So now I'm going to briefly talk about the MPS development that's been occurring at UKHSA. The Emulate system has been used to generate airway and alveolar chips for SARS-CoV-2, as you've just heard beautifully demonstrated by Tanya at Quantum Institute, but we also intend to set that up at UKHSA, and that's been funded through the University of Liverpool, FDA, and NIH. Additionally, we're using the Emulate system to generate an NHP chip for SARS-CoV-2 analysis funded through the VIS Institute, NIH, and FDA. We've also been looking at hamster tissues, which have been used to seed cultures for SARS-CoV-2 infection. That's part of a CEPI task order that we're also conducting in parallel. We're also looking at interconnected systems being designed for human lung, gut, and brain for SARS-CoV-2. That's also CEPI funded. And we're developing a system to add cellular immunity at BSL-3 to the MPS system for SARS-CoV-2 analysis so that we can eventually dissect perhaps some of the cellular aspects of immunity to vaccines in these MPS models. And of course, as other presenters have already done so, we have ambitions to widen our infectious disease portfolio for MPS for some of the organisms that I had listed in my earlier slides. And as our other speakers have also indicated, there's this wonderful MPS World Summit coming up in a couple of weeks in Berlin. And thankfully, we have 5 poster presentations, which there is a UKHSA member staff present, and I'll be happily, I'll be there engaging with as many people as possible to talk about these studies and the studies I've been hearing about today, which I've got to say are fascinating. Next slide, please. I wanted to also quickly show you what the challenges that we have in the UK. On the left-hand side, you see a flexible film isolator and what we would describe as a hugely generous space available to us within this purpose-built, commissioned, paid for and installed facility, which is now available to us. And as you can see, we can fit an isolator containing an Emulate system, a Zoe and an orb, and a fluorescent microscope, all within that space. Next slide, please. And as you can see, Kevin Naomi on the left-hand side, actively involved in a SARS-CoV-2 infection with an interconnected chip. In the middle, you see a view from the inside, from within that flexible film suit, and you can see Naomi and myself on the outside wall. And on the bottom right-hand corner, that you can see Naomi actually manipulating an interconnected system within the isolator, within the incubator of the flexible film isolator. And you see there's plenty of space in that left-hand corner slide for people to help manipulate samples that Naomi had passes to them. Next slide, please. So the ask for this talk was to identify perceived gaps from our perspective. And that's, when I say our perspective, I mean UKHSA and not from all the other speakers. From a biocontainment point of view, BSL-3, we've now created a safe working space for that. I think we don't have a gap in terms of our BSL-3 BSL capability apart from throughput. On the BSL-4 front, we have aspirations to do what the IRF have done, but that is going to be a longer term objective. And really, we need to be comfortable at BSL-3 to transition to BSL-4. In terms of safety, we have good BSL-3 compliance, but that it is complicated because it's very demanding on staff, even with a flexible film isolator. And so we need to think about the designs of these systems and consider what the restrictions are that we have to apply to, and maybe a perceived gap is there for designers to engage with us before we actually go and buy the equipment, so that they know exactly the challenges we have in the UK, which may be different from the US. In terms of reproducibility, I know I said this yesterday, and apologies for clustering the discussion with this, but I think it was I hit a note in terms of mammalian cell cultures. We've been hearing from all of the speakers, they have their much prized sources of cells, and it might be a supplier or it might be their specific private arrangements. But what we actually need as a community is to set up a global centralized sourcing capability where 3 or 4 different systems can use the same cells without there being any MTA complications or shipping complications. And I know I said this yesterday, that 40 years ago, people used to have their own colonies of mice, and that's all history. Now you go to a certified supplier and you know what you're buying because it's all been screened and genetically identified, and everybody knows what you're buying. And other people who in different facilities can use the same cells. I think this is what we desperately need in terms of being able to comply with the reproducibility requirements for a regulated study. In terms of accuracy and precision, I think accuracy, I think MPS is a more accurate reflection of what happens in humans, and I really don't like the use of cell lines unless we have to use them. I think MPS offer greater accuracy, so we don't get false negatives or even false positives. And also, from my perspective, being able to add these wonderful immune complexity that we're hearing about today is something that we have a gap in. In terms of agility, we need to be able to change what we're doing to address a specific need, and that might mean that we've got to move from one supplier to another, so that the systems we're using are fit for purpose, and that might change from both the therapeutic vaccine or drug and also from the organism that we're looking at. But we desperately need cell-mediated immunity capability within the MPS system as an animal offers us. In terms of throughput, I believe that everything I've seen has aspirations towards 96 wells, and certainly systems that we're using are very restricted in comparison to the M&A and ELISA throughputs that are achievable. But I think that miniaturization and the availability of analytical technology to help us increase that throughput is perhaps a perceived gap. Next slide, please. There's a long list of people that I'm very thankful and grateful for, but I need to mention CEPI as the funder, UK University of Liverpool as a funder, FDA and NIH as a funder. And with that, I'll stop because I know, Anthony, you're short on time today.
Wonderful. Thank you very much, Simon. A really excellent assessment of where MPS can be used to tease apart certain areas of biology, and great to see you highlight some of those challenges that we're facing, which we'll come into, I'm sure, in the next bit of the discussion with the panel meeting. There is time just for a couple of questions or so. I don't see any in the chat, but I just wanted to maybe ask, in terms of what you're saying around the need for a sort of a global centralized source for cells and cell material, what's preventing us from setting that up? I mean, I guess it's a fairly big question, but what changed in the in vivo landscape that meant we moved to that kind of system, and can we sort of accelerate that for cell-based?
I think there are a number of kind of events, and I'm not going to be specific about it, where people had been using their own colony of mice or breed of mice, and that when someone tried to replicate what they were doing with an apparently the same breed of mouse, they got a different result. And it turned out that these various inbred stocks had different MHC types, and though they were being called the same thing, the MHC portfolio within these colonies was slightly different. And so I think what we actually want is to be able to tap into the human MHC rainbow that we have, and have cells that represent each of those MHC types available to us, so that at the moment everybody's, I would say, fairly jealously guarding the cell sources that they have. And I completely understand that, because that's a critical reagent. Without that reagent, your project is going to die. But BEI over the years have been a central resource for BARDA, NIAID, FDA, and more recently CEPI as well. And that model that BEI has of having centralized sources of virus, cells, and other reagents like antibiotics and monoclonals, even monoclonal antibodies and reagents. Those critical reagents have meant that all of US funding bodies that tap into BEI are using the same drugs, the same virus, the same cells. And I would have thought that that Mark from NIH would be able to speak to this just as well as I can. But I think that if you take an animal supply and they look at MPS, they think this is disruptive technology. It threatens our business. You could say to an animal supplier, no, this is an opportunity for you. Maybe what you're doing, instead of breeding mice and rats, you can start generating cell lines and distributing in the same way that you're doing with live animals, having those resources. And I know at UKHSA we have the European Centre for Animal, we have ECAC. The WHO has now got a global biohub, and NIBSC, or MHRA as it's now called, has also been distributing well-characterized stocks of virus, and I know that those types of hubs could be used to create an international supply of validated cell lines, so that people that wanted to do a validation study could be using the same cells without being restricted by the MTAs that people use to protect their precious resources.
Wonderful. Thank you, Simon. I'm sure some of those will get picked up late. Some of those threads will get picked up during the course of the panel discussion, which we're going to move to now. So I would like to just thank all of our speakers for their presentations today. I think it's been a really stimulating and thought-provoking session. So I would now request all speakers, if you could join us for the panel discussion and put your videos on, and I will hand over to my MPS core board colleague, Candice Kerr, who will take you through the panel discussion. Thanks, Candice.
Hi, great. Thanks, Anthony. Hi, everyone. I'm Candice Kerr. I'm a program officer at NIAID in the Division of Microbiology and Infectious Diseases, and I'll be the moderator for this final session. Thank you for staying on for these past 2 days. This discussion here is really focusing on addressing some of the dynamics or parameters of MPS use for regulatory purposes. So we're going to have roughly 4 to 5 minutes to address 7 questions, which will be led by each one of our speakers. But this is really meant to be an interactive session by our presenters and the audience. So audience, please feel free to raise your hand or put your question or comments in the chat. So with that, without further ado, Dr. Funnell, would you like to start us off on the first question, which is, what types of data will authorities want to see in regulatory packages?
To start with, I guess from a regulatory perspective, regulators want to see reproducibility, they want to see accuracy, and they want to see precision. And those themes go through any data set. But they're used to seeing non-human primate data, they're used to seeing animal data, and they know that there is biological variability, and they understand that. But I think that we need to demonstrate that what we're doing is reproducible and reliable, and maybe some validation studies whereby several different institutions do the same thing to demonstrate the reproducibility of what we're doing across sites and maybe even across platforms. And maybe that can only be achieved if we're using the same cells. So that harks back to the answer that I just answered from Anthony. We can't do that unless we have a centralized source of cells, because I don't think that if we all use our own donors to test one drug against one virus, I think we'd all get a slightly different result.
Yeah, and I just to add to that point, Simon, I do think that actually that cell donor variation is actually a really important part of MPS as well, and that's actually, you know, something we should capitalize on in terms of MPS data for regulatory submissions. And I think it was highlighted yesterday about numbers of different donors that are used within studies and how that can be applied to the regulatory submissions. As a sort of, people like to use the word sort of clinical trial on chip as a sort of catch word. So I do think that's also a really important part of MPS, but I think that we should be more upfront about that when we present data, and also sort of showing the characteristics of our donors as well. I think that's something else that was brought up. I think that's a really important part of MPS work.
Yeah, I support what you just said, Emily. And it's made me think again. When we're doing mouse studies, you might have 30 or 40 mice in a study. In an NHP study, you might have 6 animals per group. In MPS, a lot of people are using the same donor, so we're not even getting the same level of difference that we would in an NHP study. So I think we need at least an N equals 6 donors for a study to give you that kind of multiple donor insight, rather than using the same donor over and over again. So I'm just adding to what you said, Emily, but I'm gonna keep quiet and ask someone else to speak about that.
And so just to continue the conversation further, in a lot of ways, we're looking at power studies and statistical analysis, determining how many of any one cell source is required. And it would be very interesting for the panel to speak on what their experiences are with that. Have they started looking at it? What have they noted? Obviously, the lowest hanging fruit are sex differences between these samples and that sort of thing. So I would love to hear from anyone in the group or the audience about their experiences in this space.
Just briefly, just to add, is that sometimes, especially in the case of, let's say, commercial donors where you're looking at primary cells, sometimes the sourcing, it's very limited in terms of the data that can be collected from the patients. Because I mean, I speak here, I don't know what it is in the UK, but in the US, there's a lot of information, medical information that is protected by HIPAA rules. So, and you can't, so there are information that, let's say, for your setting up your MPS system, you would really like to know what type of medical condition the patient or the donor is suffering, but you legally cannot ask those questions and most likely the commercial source cannot collect that information because that is private medical. And I think that is a major, I don't want to say stumbling block, but there is a limit to what we can know about our donors when setting up these systems.
Yeah, I'd agree with that point. I think getting as much information as you can from the donors is really important. So working closely with the cell suppliers and being able to understand that is really important. So just speaking from our standpoint, we have looked at different donor characteristics in terms of things like race and age. Some disease states have been able to look at, such as different mutations. So obviously we have a NASH model at CM Bio where we can look at different mutations in different donors. And that's been very interesting to really tease out differences in sort of drug efficacies and different details like that. So I think there are certain points where you can start to understand a bit more, but I think there's certainly more that could be done in the future.
Yeah, thanks, Landy, for saying that, because I found that metadata is absolutely critical to give us the diversity that we're going to need in the future. And at the moment, we're completely at the mercy of these 1 or 2 suppliers. And even when they say, yes, we have these 6 donors, it turns out that 1 or 2 of them aren't very good. And that I know Tanya's smiling, you could speak to that because but what I want to quickly say is that even when you think you've got diversity, the current suppliers don't have what our needs are in mind when they start out. And in the project that we're conducting with the University of Liverpool, we're starting from scratch and we're trying to actually put in place ethical permission to acquire that essential metadata that we need up front before we even take the sample. And that's what it needs. It needs a commercial supplier to say, what is it you guys want? And we say, we want age, sex, we want race, we want MHC type, we want whether they've got background illnesses and whether they're on any type of drug. That's a bare minimum. That's before I've even got started. I mean, I'd like to know about the BMI, whether what their diet's like, all that type of meta information. But if we go and say, this is a list of stuff we need, and then go to a commercial supplier and say, you don't do this, but this is what we want. And you compare that with a data set. When you order a batch of mice, you get the genetic integrity of the stock, you get screening for all of the known pathogenic diseases over time. You know what their diet is. That's the metadata that we get for animals. We're going to at least need that type of data for what we're doing in NPS. Over.
This is great. This is wonderful. This is the type of discussions that we really hope to continue as we move forward. And to keep to time, I apologize, I have to be the bad guy, but we do need to move on to the second question, which Landis, you can take over for us. But the question is, why aren't publications enough for regulatory submissions? And maybe you can just start out with what is and isn't sufficient based on any of your experiences, and what would potentially be the utility of combining multiple labs data to help sort of qualify, in a sense, in a general sense, the data that everyone is acquiring.
Hi, thanks for that. So I think when addressing this question in terms of, why can't you just combine publication data? And it's something that the previous panel yesterday it really highlighted and pointed out, and I kind of wanted to stop them and be like, wait, this would be great starting conversation for this, because I think, at least from my limited experience with regulatory agencies, one of the aspects that they ask is a certain level of standardization and validation of the model. And I think that even across the data, we sometimes lack. And when in terms of publication, depending on the criteria for publication in different journals can vary greatly between them. And then the cell types, there's just this lack of standardization in terms of what gets published, what information is included. And even if you go through the protocol, something very basic that, let's say, for example, the FDA asks, they need to have very specific example in terms of the protocol that is used throughout the experiment. Even that in publications can sometimes be a little fuzzy. And I think that's going to be a major stumbling block for using publication for regulatory, I guess, use. So I guess and I open up to the panel discussion, but I feel like in order for it, if you cannot use a standard cell type, for example, to test, is there a reference? Let's say one example here would be like for therapeutics. Can we have a reference therapeutic where you can compare across publications results, so that you can really start to see, is this a model? One, is this a model appropriate for what you are testing? And then how can you measure? What are the parameters to at least tell, is this a good model for this disease? Is this a good way to test the efficacy of this compound? So again, this is all to say is that there needs to be some sort of standardization in some way to validate each model for a particular use across the board. And we really don't have that yet. I think we're trying to reach that, especially through workshops like this, but it's not there. And for something to go into let's say a regulatory package, there needs to be some way to have a across the board comparison between all the different models and all the different potential uses of that model. any, I'll hand it over to others if they have any other input in terms of what their experiences have been.
UKHSA has conducted GLP studies, and the documentation required to comply with the regulatory study is enormous. And I know that publications have now started accepting supplementary data, but it's not even a tenth of the data sets that are required on a regulatory submission. And so I think page limitation is probably the major inhibitor there in terms of what you're allowed to submit into a publication. With a regulatory study, we put all of our raw data, which has been 100% checked, into the data package. And so it might be a 300 or 400 page document for a single study. And we would get a 6-page publication, if we're lucky, of that study published, because publishers don't want to see all that essential raw data.
So I guess adding on to that, so do you think that the MPS field could benefit from establishing some sort of repository database where you can have all this data, this raw data in there to reference for a regulatory package? Because
I think, do we have any regulators on the call? Please put your hand up if we have a regulator on the call. I can't see one. I'm just checking the second page. I can't see a regulator on the call. I think a regulator would want their own database, and there are digital systems that the FDA have set up for raw data. And what they want you to do is, when you've done a regulatory study, you submit your raw data to. Oh, Nicole, you've got your hand up.
Okay, so my first disclaimer is that I myself am not a regulator. However, we work very closely with regulators through ICVAM, Interagency Coordinating Committee for Validation of Alternative Methods, and the sort of most fundamental concept of what we do in terms of validation is understand regulatory needs sort of from the get-go and ensure that our validation studies and the data that's being generated is fit for those regulatory needs and contexts of use. Now, that's going to depend on the regulatory agency, the center or the office within that agency, the regulatory context of use, whether the data is being submitted for, chemical screening and prioritization, or for hazard assessment, or for risk assessment, or for something like a new drug application, et cetera. But I think that there are a lot of opportunities to work collaboratively with regulators, the FDA in particular, and EU regulators as well, have been very forthcoming in some of the webinars that we've done through the MPS core work group about encouraging submitters to reach out to them, to start a dialogue early, to have that constant communication as these studies are being designed and performed, to ensure that they are being targeted towards what regulators want to see as part of those submission packages. The other thing I wanted to mention, and Dan, if he's able to unmute, might be able to expand on this a little bit more, but exactly what Landis was talking about in terms of a database already exists. So it used to be called the MPS DB. It's now called the Biostatistics Analytics Platform. This is an effort that was vast majority funded by NCATS under their tissue chip consortium. And we funded a small part of it through this MPS core work group on developing a COVID-19 portal. And this is a platform that allows tiered levels of access, right? So you can have projects through the database that are totally public facing, and that data is public facing, or you can have it confined to just a select group of individuals, or just to yourself. So there's varying levels of sort of secure access where you can store, large amounts of raw data. And then there are analytical tools that you can access through that platform as well that are, I think really, really helpful. So I just wanted to mention those things. Thanks.
Thanks, Nicole. I think we'll go on to the next question in lieu of time. Dwight, so we put you on the spot, my federal colleague here. How can you very briefly explain how can government organizations help advance the study of viruses of pandemic potential or other pathogens?
Yeah, and I think that there are a number of things that can be done, but it depends somewhat on the agency. So Mark Williams talked much yesterday on NIAID-DMID's pandemic preparedness, and Simon referenced some of this as well. But we've gone with a prototype pathogen approach rather than a priority pathogen approach. The prototype pathogen approach would help with flexibility. There's a prototype of each major viral family that could be of pandemic potential, and if you can focus on that and develop countermeasures, then for that prototype pathogen, then it can be easily swung over to others within the family. NIH also has an antiviral drug discovery centers program for pathogens of pandemic concern to help with drug discovery for targeting conserved regions of a variety of different viral pathogens that can hopefully expedite what could be done in a pandemic setting. And I think, given the setting, I think everybody knows that MPS have been a major benefit in SARS-CoV-2. So I think especially in the setting now where we have many limitations on non-human primates, then MPS is really of interest and can be used. And the NIH does also have a human tissue bank that's been collected over the years that has not been actually utilized that much. I think once you start moving to other agencies, I think we've just talked about some of the regulatory aspects. I know that FDA is considering some of the standardization aspects of the MPS, and they actually are even developing their own assays to characterize and measure potency of cellular or biological products. And FDA, I think, will help a lot with the regulatory aspects. But then, of course, we need other epidemiological assistance, CDC, GSAID, and then the One Health program that I think would all be major contributors, because we need to tackle it according to many different perspectives.
Great, thank you. And I just want to add also that Dan from ANCATS had put in a link to the biorepository MPS, I believe. And Anthony has also put in what their agencies do to help support this research. And of course, as you mentioned, Dwight, we do have a preclinical models of infectious disease that now utilize MPS and have demonstrated the utility of it for emerging pathogens. So thank you. We're going to go ahead and move on to the next question. But if you have any questions about this, please put this in the chat. Number 4, Tanya, you're on. It's identifying a significant current discovery in an immune-focused MPS development that can help move the models for regulatory purposes. What do you think?
Yeah, so as you've seen from my presentation, my main expertise is actually in epithelium and barrier functions, so especially in the gut. So I'm hoping that other speakers that clearly have a lot more experience with adding the immune component will be able to contribute their perspective on different models, etc. But I will start with an example on gut on chip where immune cells were added to build on the complexity of the model. And this was recently done and shown by the Emulate with their IBD model. So they've used very well characterized colon intestine model that they primed with IBD relevant cytokines. And then introduced peripheral blood mononuclear cells into the vascular compartment, which then, with the help of basically the inflamed system and chemoattractant molecules in the apical site manage to then recruit the immune cells into the epithelium. So they've extensively characterized this model and looked into different aspects of the disease pathogenesis, and they validated it against clinically relevant therapies to demonstrate efficacy and mechanism of action. And they've Emulate also invite other labs for beta testing to just test their protocols, which is we were one of these labs to be involved to provide feedback, etc. And I think that all in all, together, this approach really then provides kind of reassurance, validity to really facilitate regulatory acceptance for this, for drug development, drug screening in this case, for this model for the IBD. Yeah.
One thing I wanted to mention on this, I think, with regards to the immune component, is it's really important when we're thinking about primary cells. I think Simon might have mentioned this in the cell chat earlier about matching. So immune cell matching, at the very least, HLA matching with the epithelial tissue, or whatever tissue you've got in there, endothelial tissue. I think this is a really important part that sometimes is skimmed over. And I think it's something that needs to be continually considered in these studies. I think it's very, very important.
It's probably very difficult to get all cell types matched for HLA type, but it's something definitely that we should aim for.
For sure, yeah, and I think it harks back to what we were saying about, you know, having those relationships with cell suppliers or having a centralized bank, because then that allows that matching to occur. I know I've had numerous discussions with cell suppliers about matching, particularly about immune matching, and they are very open to it, and they understand the need. So I think it's something that hopefully, you know, as that technology progresses, the primary cell / iPSC technology progresses, MPS technology can progress with it, as it's the basis of what we do.
And Emily, maybe that's sort of a segue to our next question, which you're leading. So it sounds like a natural progression into the next question, which is what should MPS developers focus on in the next 5 years and MPS technology or research to be most effective for infectious disease research?
So a lot of what I was thinking about for this question actually has already come up, which is great. So standardization, cell sourcing, etc. But I think something that MPS developers should really be working on is actually the trust and understanding actually of MPS technology. I think that's something actually in the wider field that is really lacking. Obviously, you know, when we're in our MPS communities, we talk about it a lot, we all know what we're talking about, but actually when you go into the wider community, I don't know, for me, if I go to a respiratory focused conference, actually not that many people know about MPS yet. It's still, although we've had the FDA Modernization Act, and you know that's been a really big deal in our field, actually communicating that further still needs to happen. So I think MPS developers can really be a part of that alongside, you know, all the other partners. So I think that's really key, first of all, as a sort of subset to the development. I think in terms of the development, actually, something Simon mentioned in his presentation was developers really need to think about the application of their hardware into the BSL-3, BSL-4 lab spaces and the future design. I think that's something particularly in this space that needs to be considered. And also thinking about the general requirements from the field, because obviously there are going to be different requirements between academia to pharma, and really understanding what people want. I think there's a whole influx of different models, chip types, outputs that are currently out there, and a fantastic amount of work that's gone into it. But actually getting that feedback now from people that want to use these systems is crucial. So things like, you know, what clinically relevant markers do pharma companies want to see, or even the regulators when they're looking at submissions. You know, what organ models are actually important to people? We've talked about different infection models. So HIV, I think, in particular, was really interesting from Dwight, and really understanding actually the different cell types that might be needed, and the different target organs versus sort of secondary organs. So I do think that that two-way feedback between MPS developers and users is really, really crucial. So I don't know if anyone else has got any.
Can I just jump in there? If you look and see what's happening in the in vivo world, there's a bit of a revolution going on in terms of telemetry, and telemetry is revolutionizing what data you can acquire from an animal while it's live. Instead of having to go in and look every 4 hours to see, you know, what the temperature is, or how they're moving, you can do this with an implant and get that 24/7 data. And that's a technological intervention, which is a refinement, because then you don't have to go and disturb the animals every 4 hours, 24/7. And so if you then try to say, well, when I know Tanya's work really well, and she's done a fantastic job, but what we've had to do is we've had to go and sample every at time intervals, take the sample out, think about how we're going to get it out of containment, preserve its integrity, and then analyze it at BSL2. That's laborious. But what we actually need is the microfluidic technology to be able to get those cytokine and PCR measurements done without taking a sample and having to take it out of containment. That would revolutionize the technical challenges that we have in trying to get the data we want without having to go through the rigmarole of taking a sample and getting it out of containment and decontaminating it and maintaining its integrity.
So can I piggyback on that, Simon? Do you think the technology is already out there, but it's the placement into this BSL 3-4 scenarios that have been most challenging?
So that's two questions. I think the first question is the technology there? I don't actually know, because the biofluid, in diagnostics, microfluidics is really progressing at an enormous rate, and the miniaturization that's occurring in diagnostics is amazing. But I don't think that we've got an interface with those people that can do that kind of microfluidic technological detection. So the answer to the first question is I don't know. But I think if it was available, we would be keen to get hold of it, because it's going to reduce the amount of effort and risk in doing a high containment experiment in terms of the UK, in terms of the restrictions that we have in the UK. Tanya, do you want to say anything more about that? Because you're the person that's been on the coalface with that.
Sorry. Yeah, that was actually one of the challenges that when you actually left with, yeah, let's go, you don't have a separate plate reader in CL3, you might not even have the space in the lab, right? So that's the fact. But some technologies are better suited, for instance, for imaging, and I know that there are different platforms that are perhaps more suitable for on-chip imaging, and some are more suitable for sampling, and so it really depends then also what you want to do downstream, etc. But it is challenging, I agree with that. But like in our case, we had to then get another kit to then have one in CL2 and one in CL3, and it's not cheap, you know how it is. So you have to in some way either double the amount of equipment, or just think about how you get around this by using appropriate methods to deactivate the virus. But so far we have, I mean, with this limited experience that we had in that respect for this experiment, we managed to do the desired downstream analysis that we wanted to. So, but if it can be done easier, then that's welcome.
Gabriella, you've done it at CL4, so you must have a view on this.
Yeah, I do. I think it depends what samples and what is really collected. And I think I mean I can speak to our experience, but I think it will heavily depend on the institute and that institute's sort of SOPs and standards and work practices. But yeah, I think what you just mentioned, it will kind of go into the last question. In terms of triggers to treat and having actually readouts that can be quickly accessed. But yeah, hopefully, I guess the goal would be to have protocols in place that would make these readouts easy and rapidly accessible. And so I think that would just mean that having more of these readouts back in containment would just be a requirement.
Great, thank you. And we'll hear more from you in just a minute. Question number 6, Saeem, if you would mind spearheading this one. It's where within infectious disease research are the best opportunities you feel for reducing or replacing animal use by adopting these MPS approaches? Maybe you can just sort of highlight your experience.
I think these microphysiological systems have a good opportunity to replace animal models in certain areas, like for drug screening and efficacy testing. They can combine they can combine those things. Like, for example, when we were trying to do test the intestinal tissue model, what we did was we collaborated with AstraZeneca, and then they had basically what they want to see is the model. Is the model working or is not working to replace the animals? That's what they were waiting for. Then we said, okay, if we want to test that type of concept, you have all the human clinical data collected for certain drugs that has been that has passed the animal experiments but failed in human clinical trials. So they collected some of those drugs which was failed drugs, but they have passed the animal. They were clear with rats and dog experiments, and now they gave us those samples to test. And when we test those samples on our tissue model, then we said these are problematic drugs. They were blinded, and we told them these drugs are problematic. Then they said, okay, and we asked them, do you have clinical data? Can you correlate these things to the clinical data that you have? So alone, the developers of these microphysiological systems cannot bring these things by themselves alone. There should be industry collaboration. That's one thing I can see if they want to see this type of drug, because these systems can work for early on in the drug development process, for example, to cure a certain type of disease. So they can have value in the early on, or they can have value for investigative drugs to study mechanisms, why they failed this type of drugs, and so they can make some kind of improvement in that area. That is one thing. So there should be a collaboration between the industry and the developers of these platforms. That's one thing that we have to consider. We can study disease transmission and so on, but the organs that are going to be connected to each other, we have to be clear which organs are going to connect to each other for a certain type of disease. Like, for example, these viruses are receptor, basically they use cellular receptors. So like if you take HIV, you don't have any another animal model. SIV is a different thing, but for HIV itself, there is no animal model. So if we are going to model it, probably we have to concentrate the reproductive organs, like the vaginal tissue models, with that of the immune cells. Those are type of things that the concept has to be developed, and the data has to be generated from that type of connective tissue models. Otherwise, we may not answer the right questions. We can use these models for personalized medicine also. To develop personalized medicine, probably drug resistance could be there. People are different, so by putting these individual cells from donor individual patients, you can develop microphysiological systems and test some effective drugs. But the whole thing lies in this case is because from our experience, what we have is we have validated models that in OECD, but it's not yet here in the United States. Those models are not approved here. But on those validation models, it takes a long time also. So there should be some mechanism to bring the regulators and the developers together so that at least in a short period of time, decision can be made if there are convincing results. So protocol, as they are now, protocols are not developed and standardized. That's one issue that it has to be. There are about 55 or more microphysiological platform developers, and each protocol is different. Each one are different. So there should be mechanism to consolidate those type of things. And then there is no universal media that we use to bring these two different tissues together. Otherwise, it's no different from the static models that we have. Individual models can have individual media, but we have to. There should be some development in universal media to access to, like blood, for example. Blood is our universal media in our body, but equivalent to that, we need some kind of media development. I think that's forgotten. So the platforms are not validated for us to, for example, for the ocular test. I can give you some example. The ocular irritation, eye irritation test, the DRESS test was developed in 1944. And then the test methods, the in vitro test methods were in 1984. And for approval, it takes another 27 years. So that's how the regulators are working. So there should be some kind of system to enhance it. In one way that we do when we do validation studies is that there should be lab-to-lab comparison. So certain chemicals, certain key chemicals has been selected, and they should be studied in one lab and in another lab, and the protocol has to develop for those, and there should be reproducibility, reliability, predictivity. Those type of things has to be there. Then I think will the fit for use concept, convince the regulators? I'm not sure at this point, because they may, because there is liability and things like that, they are afraid to touch it. So there should be, the burden is on us to convince them these things are reproducible. These things can do we have to define the endpoints to measure what are the endpoints that we are measuring for each type of study. Those are the type of things that are essential for to bring these ones to replace animal models.
So thank you very much for that response. And you can see in the chat, you've addressed a couple of questions from Dr. Dawn, and Dan has also reiterated exactly the types of comments that you've made here. So that's been very helpful. If we can, and I apologize that we're already 15 minutes over, which is, so please hang on to the last and final question. The final punchline Gabrielle is going to lead us on to is how can the PI or regulatory authorities define or justify this trigger that signals the start of treatment with countermeasurements, and how can they use MPS to do that?
Yeah, thank you. I think that's a very good question, and actually a hard one too, because I'm not sure that we have that quite figured out in animals either, at least to some extent. But I think there are actually easy trigger to treat or signals that would indicate that a treatment is needed, and that could be a certain time point in the experiment. So we're going to start treatment at 24 hours or 6 hours post-exposure. But that I see that more as a experimental protocol and not really a trigger to treat. If we want a real sort of physiological trigger to treat that is based on a response, then we need to look at things that are physiologically relevant. So I think I can draw a lot from the animal models. So when we have an animal model for, let's say, an infectious disease, we can either do observations, so clinical observations and then just draw clinical data to see, how is that animal doing. As Dr. Fano mentioned, telemetry is one option. So this can range from easy, just temperature implants that record the core temperature, and there are more complex ones that can measure the animal's blood pressure, so we can identify hypertension or hypotension as a trigger to treat. And that data is available instantly. And then, so I would look at that data. So for any trigger to treat, we need two things, the way I see it. We need data, and then we need a rapid readout or a rapid test, or something that will tell us what that data means. And so if we look at animals, we have different sort of tiers of data. So we will have the clinical, which is available right away. We will have maybe telemetry data. They're also available instantly. Then sort of on a second tier, we can collect blood. And then that blood, we can do CBCs, coags, chemistries, which doesn't take much time. It should be available on the same day. And then on a third tier, on a more complex manner, we can look for either viral load or bacterial load and identify that as a trigger to treat. And so I want to sort of like put that in parallel to chips. So when we have MPS or organ chips, we can, when we just look at them under a microscope, we see the morphology of a chip. So perhaps if we see a certain, fluorescence, or if we have a certain morphological presentation, that's available right away, and we can start treating that particular chip. And that doesn't mean, I think that's a natural way of looking at it, because we don't really tinker with either the chip or the virus in that sense. Then on a second tier, we can collect effluent and test that effluent for the presence of cytokines, the same way we would test blood for certain markers. Again, that would take time. Again, passing the effluent out or inactivating it is one option, or just doing the analysis real time. And then on a third tier, we can test the effluent or cells for the presence of virus or bacteria or toxin, and have that as a trigger to treat. So I think the physiological relevance is important in identifying a trigger to treat, and then also the availability of a test or feasibility of having a quick readout, so we can actually use it as a trigger to treat immediately. And I think it's a complex question, because it will depend on, it will be organ specific, and it will be condition specific. So by condition, I mean either a toxin or a virus or bacterium. And for example, from a virology perspective, we thought about this, how nice it would be to just have a EGFP tagged virus, and you just see fluorescence as an indicator of replication, or you could have a low bit encoding transgenic mouse and a high bit encoding virus, and once they come together, you use bioluminescence to identify that there is replication going on. But then in this instance, you would then maybe change, by changing the wild type virus, you would change, the wild type or the virulence or pathogenicity maybe in some way. I mean, that's something that a reviewer might say. So I think these are all considerations. I don't really have many answers. I just have more considerations or questions, and I hope others have input on that.
Yeah, no, I think that's great. A lot of food for thought. I think we could have a whole hour on this conversation. You have lots of wonderful ideas you just put out there. And Simon, you had your hand raised first.
I agree. That was a really comprehensive answer. And I just wanted to add that a number of speakers today have mentioned transepithelial resistance and measurements of resistivity of the monolayers. And I know that's something that's very specific to the viral pathogenicity, and that's something that could be measured very easily within the well. So you could have a 24/7 readout of resistivity, and when it reaches a critical point, you know that this is cytopathic effect. This is, as Tanya really eloquently described, a derangement of the normal junction between the cells, and it's an easy thing to measure. But that's a specific clinical sign that would be analogous to an animal having a clinical observation of illness, and so it needs to be specific. And it wouldn't be, I don't think the regulators would accept a time as a trigger. You're going to need to see something specific, and I think that resistivity could be added in as that list of things that might be able to be available in a microfluidic format in the future one day. Over.
And Landy, your hand was raised next.
Thank you. Simon kind of just took the words right out of my mouth in terms of some of the things that I did want to highlight with these MPS system, which are, one of the benefits of the system is the inline real-time sensing. So you can sense during the course of an infection something like ROS production or other immune or cytotoxic potential effects of these cells inline on the tissue. And also I want to point out that given that these are primary human specific tissue, you can look for biomarkers of certain disease states that could now then be applied in the clinic. So you can be looking for things like volatile organic compounds that the patient might be releasing, that it's an indicator that the disease is progressing. So there are many different options available that MPS system, especially if you have integrated sensors within these systems that could then be applied in the clinic or later on, once validated, that can act as triggers that to alert the doctors in terms of how the disease is progressing. And I think that's, again, one of the strong points of using these human-specific rather than animal models to really kind of bridge, to kind of act as an alert system for the doctors that the patient is either progressing poorly and needs some sort of treatment.
So to continue that just a little bit further, thinking of it in another way, how do we know the manifestations we see on that MPS matches your first trigger to treat in the patient? How many viral particles would you need in a nasal epithelium to know that your patient is now at a point where it shows symptoms, for example, that you would then lend to that sort of treatment? How do we think about these things? How much do we know about how and when the MPS mimic the infectious disease process that we can utilize it in a patient? So I know I just kind of switched the question around a little bit, but you also thoroughly covered the other question.
Yeah, that is a very good point, and so difficult to answer. I think we would. I think what could be done, and maybe hasn't been done really, is to really first understand the model very well, and based on retrospective data that's combined and looking at a specific trigger to treat or a manifestation of it or a readout, and then take another bulk of MPS data and analyze it and then just see kind of what correlates. I'm not sure if ever anyone has done that or is interested in looking at that, but I know maybe others, I see some hands raised, so maybe others have ideas.
And should we go with Landy first? She's first on my screen. I apologize.
No, I just wanted to add, I think this is one of those scenarios where it would really benefit if researchers had good connections with, let's say, hospitals or research hospitals where you can establish those types of collaborations and start making those sorts of that sort of data that you need to really bring MPS system into kind of like a more, I want to say more of like a diagnostic or disease state diagnostic. So I think that's something that I think as a field, we really could benefit from having that strong collaborative connections with the medical field.
Emily?
Yeah, I think that's a really good point. Echo that completely. I think the other thing is as MPS developers, we're very used to scaling. So that's something we do to develop our models, and that's something we should also be now applying in the same way. And we've done that a lot with looking at things like drug efficacy and toxicity. That's something we do a lot of, is that scaling activity, and that should now be applied to infectious disease. I think also, as Andrew said, having that two-way conversation, again, talking about the two-way conversation between, yes, hospitals, also pharma, getting that clinical data, feeding that back in, using modeling, mathematical modeling as well, is something we see as really, really important, and we've done a fair bit of as well. And I think all of that together really should allow that relationship between the trigger and treatment, and then applying that back and forward between the clinical data. But I think that can only be done when combining it both ways.
Thank you. Simon?
Candice, you did switch it around, and I think when you said, how do we know what the triggers are that we need to use, it may be, and I see that there's a question from Xishang Dong at 1647 about this. There's huge natural history studies that have been done in non-human primates for many infectious diseases, and I think we can pivot that data by generating animal MPS. And I know that sounds counterintuitive, but if we can show that the natural history that we see in a primate from studies that have been conducted in the past are reflected accurately in an MPS system, then that will give us confidence in taking the triggers that we're going to use in the human systems forward, because you won't be able to do those beautiful natural history studies that have been done in primates and other animals in humans, because you can't take a serial sacrifice of humans every day after being infected with a select agent. That data just doesn't exist, and you'll never find it, but you will find that in the archives of NHP natural history studies. So regrettably, I think the answer is to generate an NHP organ on chip, and that, I think that's the answer to the difficult question that you posed, Candice, and that's what we're trying to do.
Thank you. And Landis?
Kind of just wanted to add, because it was I think it was Emily that brought up the point that in order to get some MPS system into, I guess, the regulatory framework, is this trust, this idea that you need to establish. And I think having that, exactly what Simon pointed out, having like a non-human primate model that has that history behind it, I think that also will contribute from previous conversation in terms of getting MPS system in place in terms of the regulatory framework.
Nice. Go ahead, Gabrielle.
So I was just thinking in terms of having a non-human primate on a chip. I think I wanted to like push that idea a little bit further, because with the non-human primates, we will still have the issue of having different donors or a different non-human primate. But I wish we could design somehow, let's say we have a SARS-CoV-2 experiment in non-human primates. What if we could design an animal study protocol in a way where we could collect tissue or do sort of a bronchial alveolar lavage sampling of samples, or obtain sample and tissue ex vivo from that animal, let it heal, develop the MPS from that animal, and then put that animal on study, and then sort of do that in parallel. So have that experiment's animals specific chips and have those two things run in parallel. That would be like the most ideal side-by-side comparison.
That's a great idea, but a lot of the data already exists. And perhaps rather than having to repeat those natural history studies, what we might be able to do is go back to the colonies that supplied the animals and say, well, this is the same family, this is the same colony, this is the same gene pool, and that's going to vary from study to study, because each study will have its own NHP supplier, and they still have those NHP suppliers. And I can speak for you, could just say, we can go to our colony and and select animals that have been used in a natural history study and say, well, this is still the family. This is the granddaughter of the animal that was used in that study. And that might be a way to fix that having to do it again problem by going to different colonies and comparing colonies, because each of them will have their own MHC haplotype profile.
Right, or take, for example, if there was a banked sample of cells from each NHP that was used on study, and being able to take that material and create an IPS or something, pluripotent stem cells, and then use those to make different chips from that NHP would also be sort of like on my wish list.
There was a lot of discussion yesterday about the merits of pluripotent stem cells, and I love that idea. And I know that Donning, but in particular, was questioning whether that strategy will give you the same epigenetic profile that you get from a donor. But I think I still think that stem cells may be an answer to some of our questions once the technology is mature enough for us to be able to trust it. And I know that within the UKHSA, within the UK, blood donors can provide stem cells to patients who need them, and there are stem cell transplants happening all the time. But I wonder how many of those stocks of stem cells have a kind of use-by date, and we could perhaps access them on the day after they've expired rather than discarding them. Over.
That's a good point. Dwight, did you have a comment to that?
My comment actually backtracks a little bit, because I think Simon and Gabrielle and everyone had great comments, but I want to comment, putting on my clinical hat, because I'm also a clinician, and historically we've used like Simon mentioned, we use clinical triggers of fever, how sick the patient is, in order to guide when we would start a countermeasure. However, what's transitioned over time is that the clinical field has moved more and more towards biomarkers. And there's a few reasons for that, with differences in physical exam clinical skills, but also in increasingly immunocompromised population, some clinical signs are becoming less and less reliable. And if you look at a number of different predictive scores on risk, then biomarkers tend to have better predictive capability and help to optimize. And even now, we have an initiative for development of biomarkers to detect different stages of TB versus, say, infection, incipient infection, disease, and in trying to define where in this TB spectrum that you are, because your treatments would depend on what stage you are. So trying to bring that together is, I do agree, you want to correlate with clinical colleagues, but also the better you can define biomarkers that will help you identify where in the disease process or infection process you are, I think that would help guide infectious triggers.
That's a great point. So with that, I think we could probably wrap it up. If Anthony, you want to give us our final comments? Thank you all very much. Very informative.
Thank you, Candice, and thank you all of our speakers and panelists today. I mean, that was a really interesting discussion. I think it's thrown up a lot of food for thought for the board members of MPS Core, but also more widely the community to think about and how we can move some of this forward. So I won't keep you long. I appreciate it's been a long but very stimulating couple of days. That's really highlighted the broad applicability of MPS systems in a number of different disease areas and organ systems, but also I think importantly some of the challenges that we still face as a community that we need to address to be able to really move this forward and really realize the full potential of these models. So like I said, as a board, we're going to go away, think about that, and look forward to coming back to this group with sort of next steps and what we want to do next. But that's also, we still want to hear from you about what that looks like. So, if this has stimulated certain ideas for yourselves, do please get in touch with us. You've got our details, either myself, Nicole, or Amber, or any other of the board members. Please do let us know your thoughts and how we can help you as a community move things forward. So I think I just want to finish with a few thank yous. So firstly, all of our speakers, thank you very much. Really fantastic set of presentations, keeping to time as well. Thank you to all of the audience for sticking with us and for your participation and engagement in this. Thank you to the MPS core board members for helping us put this together. And very importantly, thank you to Amber and the rest of the NIH AV team as well in making this such a really successful event. I do just want to offer last final words to Nicole, if Nicole wants to say any sort of further closing remarks, as well as my co-chair in this.
Thanks, Anthony. Just echoing everything that you've said, and just huge thanks to all the participants. It was just an absolutely tremendous 2 days of presentations and discussion. I've just put the link in the chat as a reminder that the recording will be available online soon. And again, thanks to all of our support staff. Really outstanding job as usual.
Fantastic. Okay, well, thank you very much. And I look forward to seeing some of you either in Berlin or potentially even at the World Congress in Niagara in the not too distant future as well. So thank you, everybody. Take care. Have a wonderful rest of your week. And we look forward to being in touch soon. Thanks, everyone. Bye-bye.